Hello, everyone. A very warm welcome to first class of ophthalmology online class. A very warm welcome to all my doctor friends there. I'm Dr. Anuradha Dhawan, your ophthalmology faculty, and from today onwards, we will be starting our journey of learning ophthalmology together. So before we start, just kind of a little bit of introductory things you can say. All of us, we know ophthalmology is is the subject in which you get about 15 to 18 questions in your exam. It has been coming. And it is one of the very scoring subjects. And I hope, by the end of our classes, we will be pretty confident that yes, we can do those MCQs, which can help us crack through our exams, isn't it? So this is our common goal. Yours and mine common goal is that we are going to learn ophthalmology in a way that it's going to help us solve all our questions in our exam. Okay, so here is you can contact me for any queries on my email, through Telegram, or message in your app groups. Okay, so I'm ready to clear all your doubts or anything. You can message me during the class also. I'll try to cover it up, or after the class, so that any query which we do have, it can be solved on time. Okay, and we are going to learn with the help of our workbooks, and I'm sure all of you are ready with your workbooks. Now, how to get maximum out of the classes? So just few points there. Now, whenever we start a new subject, first thing is starting a new subject can be a little bit overwhelming. Overwhelming in a sense that when you start a new subject, a lot of new terminologies associated with that subjects will be getting into our ears for the first time. So don't get overwhelmed. Try to build the concepts. My aim is always to build a concept around a topic, so that once you understand that what this topic is or what this disease is, then you can recall the or memorize the important points related to that very easy. Okay? So try to build a concept around the things. The workbook has been designed in a way that you don't have to write too much. So my effort has been that you don't have to spend too much of time writing. We will be writing along with where some addition is needed. Okay, but at the same time, put all your focus, friends, put all your focus into understanding, into what I'm saying, into going through the workbook together. Okay, so to make best use of the workbook, get some highlighters, some pens to highlight the important points or underline the important points, which I will be telling you along with. Okay? So at each point, I will be explaining, I will be letting you know what are the high yield points, how the questions they can come through this that particular thing, so that we are able to kind of learn in a very wholesome manner. So I think with this, all of us are now geared up to start our ophthalmology. So I, Anuradha Dhawan, once again welcome you, and let's begin with the first topic in ophthalmology, which is the basic anatomy and the physiology of the eye. Basic anatomy and physiology of the eye. Be ready with your workbooks, and friends, let's move on to the first chapter. Let's move on to the first chapter. Please come to the first chapter, that is anatomy and embryology of the eye. Please open your workbooks. open your workbooks and let's begin with the anatomy and embryology of the eye. Now, in this chapter, in anatomy and embryology of the eye, we are going to discuss the gross anatomy of the different structures, okay? And the specific anatomy related points will be dealt with in the respective chapters, okay? Like, when we are doing the cornea, we will go in more depth anatomy. Here we'll be doing a basic or the gross anatomy. Okay? Now, coming to the eye, all of us we know that eye is a sensory organ. Okay? Now, the anatomy of the eye is designed in a way, what is the purpose or function of the eye? The function of the eye is to see. So the whole anatomy of the eye is designed in a way that it can fulfill its function. Okay? So that it can sense the light coming through the different objects so that an image can be formed. So that's the purpose. Eye is a sensory organ. Now, where is the eye placed in the body? All of us, we know the eye is placed in the body in your bony socket. Okay. Now, this bony socket is called as orbit. The bony socket in which the eye is placed is your orbit. So you see here, this is the bony orbit here. And this is how the eyeball is placed in this bony orbit. Okay. Now, in the bony orbit, besides the eyeball, there are few other structures also. Okay, so this figure is just telling you how the eyeball is surrounded by the other structures in the orbit. More importantly, what it is showing here is extraocular muscles. Okay, so what are you able to see in this picture is extraocular muscles. Extraocular means which are outside the eyeball. Ocular means eye. So extraocular muscles which are outside the eyeball. We'll be discussing them later on. This is just to give you a gross overview that how the eyeball is placed. So you can notice the eyeball is here. And then here we can see 6 extraocular muscles. I'm just going to name them at this point. Don't bother about remembering them at this moment. We'll be doing it later afterwards. Okay. So these extraocular muscles, as you can see, there is lateral rectus, medial rectus, superior rectus, inferior rectus, superior oblique, inferior oblique. So this is just giving you an overview how these muscles are surrounding the eyeball. And then this what you are seeing is your optic nerve, because optic nerve begins at the eyeball. This is your optic nerve. So here you get an idea how the eyeball is placed in the bony orbit and how the muscles are surrounding this eyeball and how the optic nerve arises from the eyeball and then it goes further in the visual path. Okay. Now, when I am using the word eyeball, so it tells you that eye is kind of a ball shaped. At the same time, it's not a solid ball. Okay, it's a cystic structure. It's a cystic structure. Cystic structure means it has wall and it has cavities. Okay. Now, just some gross parameters. The anteroposterior length of the eyeball, the anteroposterior length of the eyeball is called axial length. Anteroposterior length of the eyeball is called axial length. Okay. And average axial length is 24 millimeter. An average axial length or anteroposterior length of the eyeball is 24 millimeter. Okay. Now, this is important to remember because if the axial length, AL, axial length is less than average, so it means eyeball is smaller in size. Now, these small eyes are usually hypermetropic. We call them as, you can write here, hypermetropic. So if the axial length is less than the average, it means it is a small eye. And small eyeball is usually having hypermetropic, which is a type of refractive error. Okay. And if axial length is more than 24 millimeter, that is more than average, then we call it as myopic. Then the person will have myopic refractive error because it is a long eyeball. Okay, so average anterior-posterior diameter is 24 millimeter, which is known as axial length of the eye. If it is smaller, it will be a small sized eyeball. It will be called hypermetropic. If it is bigger, it will be a longer eyeball and it will be called as myopic. Okay. Average volume of the eye is around 6.5 ml. Okay. So now you imagine, you know that eye is a ball. It's a cystic structure. Now, what are the walls of the eyeball? The cystic structure means outer walls and inner cavity. So what are the walls of the eyeball? Now the eyeball walls, we call them as coats of the eyeball. Okay, we call them as coats of the eyeball. There are 3 coats of the eyeball. Okay, fibrous coat, which is outermost. The outermost coat of the eyeball is known as fibrous coat. The middle coat of the eyeball or the middle layer is known as vascular coat, and the inner is known as nervous coat. Okay, so the outer side, the wall of the eyeball, the outermost is your fibrous coat, the middle one is your vascular coat, and the inner one is your nervous coat. So let's see how these coats are placed. Come to this figure, beta. Come to this figure. Now, what we are doing right at the moment is we are trying to understand that how the eye looks like, how the eye is made up of. Now, when you see the eye from the outer side, what you are seeing is the outer coat. Okay, so here you are seeing the outer coat, but it has different parts. The whole wall of the eyeball has 3 parts. Okay, so this is the outer coat, this is the middle coat, and this is the inner coat. Okay, just for your reference here, outer coat, which is fibrous, middle coat, which is vascular, inner coat, which is neural or nervous coat. Okay, so they extend from the front part of the eyeball to the back there. Okay, so you can see they extend from the first part to the back there. Okay, now if you look at the outer coat here, now outer coat has 2 parts. It has 2 parts. Anterior part is known as cornea. Okay, so this is your cornea. The anterior part is known as cornea. Look at the figure first. Everything is written there. First of all, make a concept how different layers of the eyeball are organized. Okay, so the outer part has, in front there is cornea, and then at the back this is sclera. So sclera is going till back there. This part, this whole part is going, and it is ending at the where the optic nerve is coming out from the sclera. Okay, where the optic nerve is coming out of the sclera. Okay, so this is your fibrous coat. This is your fibrous coat. Got an idea, beta? Now let's see. So come down here. Fibrous coat is the outermost coat. Fibrous coat is the outermost coat. We have seen that the anterior 1/6 is made up of cornea. Posterior 5/6 is made up of sclera. Okay. So 2 parts of the outermost coat. Anterior is cornea, posterior is sclera. Now cornea is a transparent structure. cornea is a transparent structure. And that is the main refractive surface of the eye. So whenever the eye strikes, the light strikes the eye, first of all, it passes through cornea. So cornea is the main refractive surface and it is transparent. Okay. Sclera is opaque. It appears the white portion of the eye. The white portion of the eye is your sclera. Okay. Now, the junction of these two, please write down here. Please write down junction of cornea and sclera. Junction of cornea and sclera is called limbus. Please write this term. The junction of cornea and sclera is called limbus. Okay. And what is the important point regarding limbus? This area, it has stem cells which are called palisades of Vogue. Okay. So junction of cornea and sclera is called limbus. Limbus has stem cells which are called palisades of Vogue. Okay. So sometimes this can be asked as a theoretical question, where the palisades of Vogue are present. They are present at the limbus. Okay. So this can be your theoretical question, that where the palisades of Vogue are present, they are present at the limbus. And these stem cells we will be doing in the cornea also. They produce the corneal epithelial cells, just for the information here. So I hope you have added this point. Junction of cornea and sclera is known as limbus. Limbus has stem cells which are called palisades of Vogue. Okay. So you get an idea. So if I show you here, which part will be the limbus, beta? Which part will be the limbus? This is limbus. This is limbus. That is, this transparent structure is cornea and opaque structure is sclera. So the junction of this whole area, the junction of cornea and sclera will be known as limbus. All right. Okay. Now coming to the middle coat. Now coming to the middle coat of the eye, which is also known as uveal layer. Uvea. The middle coat of the eyeball is also known as uveal layer. It has three parts. It has three parts. Okay. Now these three parts are iris, ciliary body, choroid. Iris, ciliary body, choroid. Anteriorly in the front iris, then ciliary body, then choroid. Okay. So let me show you in the figure and then we come back here. Let me show you in the figure and then we come back here. So to make a concept of the uveal layer, come back to the figure, beta, 3 layers of the eye, figure 2.2. To make a concept of the uveal layer here, now this, in the beginning, from the front, this is your iris. Okay, this part is your iris. This is the iris. Now, iris, as you can see, has an opening. The iris has an opening. This opening is known as pupil. So just concentrate on the figure and listen to what I'm saying. This is your iris. This is your iris. And iris has an opening which is known as pupil. Then behind the iris starts ciliary body. So behind the iris starts ciliary body. Now this part, this whole part is which I am kind of blackening now. This part is your ciliary body. And as you can see, the ciliary body has two parts. The one which has got these finger-like projections and one which is the plain part. Okay, the one which I have blackened, this is your ciliary body. It has two parts, one which has this finger-like projections. This is called pars pleccata. Everything is written there. And then it has got a smooth part, which is known as pars plana. Okay, so this is ciliary body. And when this middle layer goes behind from here, let's mark it with some other color here. I can use the green color. Now where it now after the ciliary body, the middle layer continues as choroid layer, the one which I have marked with the green in color. Okay. Now why I am stressing here at the anatomy beta, because when I talk about the diseases, if you know the anatomy, then you will be able to make a concept of the disease, symptoms, signs very easily. Okay. So this is the middle layer. Middle layer has three parts. In the front is your iris. Iris has an opening in the center, which is known as pupil. Okay. Then this middle part is known as ciliary body. Ciliary body has two parts, one pars pleccata and one part is known as pars plana. And where the ciliary body ends, this middle layer then continues as choroid. Okay, right? Now let's see. So in the vascular code, come to the notes now. The in front is your iris. Iris has pupil. Okay, so iris is a diaphragm-like structure. Okay, it's like a diaphragm. Like it's like a diaphragm, and in the center there is an opening which is known as pupil. So this is a diaphragm of the iris, and in the center there is an opening which is known as pupil. Okay. Then the second part is ciliary body. And as I told you, the ciliary body has two parts, pars pleccata, which has projections. These projections are known as ciliary processes. Pars pleccata, which has finger-like projections, these are known as ciliary processes. Okay. So these ciliary processes, they secrete a fluid which is known as aqueous humor. Please write down. The ciliary processes, they secrete a fluid which is known as aqueous humor. The smooth part of the ciliary body is known as pars plana. Now the pars plana is an area through which we enter posterior segment of the eye, posterior segment of eye for surgery. Like, if you want to do retina surgery or the surgery in the posterior segment of the eye, then we enter through this posterior area, which is pars plana. Okay, so ciliary body has two parts, pars pleccata, pars plana. Another thing, please add here, ciliary body also has a smooth muscle which is called ciliary muscle. So in the ciliary body, it has a muscle which is known as ciliary muscle. Okay. We will talk about ciliary muscle in accommodation. I will tell you what is accommodation later on. Here, just concentrate on the anatomy part. So this is your middle layer or middle coat of the eye, which is also known as uveal layer. It has three-part iris. Iris has a central opening called pupil. Then comes ciliary body. Ciliary body has two parts, pars pleccata and pars plana. It also has a smooth muscle, which is called ciliary muscle. Okay. And then the third part is your choroid, as I told you in the figure. Now, choroid is very vascular structure. Lot of blood vessels are there. It is very vascular structure. And another point to know about choroid is that it also supplies nutrition to outer layers of retina. It also supplies nutrition to outer layers of retina. So you are clear about the uveal layer beta now? Okay. Now coming to the innermost layer, that is the inner coat of the eyeball. Come back to the figure here. Come back to the figure here. Now the innermost layer is known as retina. The innermost layer is known as retina, and here is your innermost layer. It is also known as neural. Now retina starts from here. Okay, so retina starts from here and it goes till the optic nerve. So do you see here, beta, this neural layer? It starts from here and it goes till the optic nerve. Now where the retina starts, you can label here this part. Where the retina starts, this part is known as ora serrata. This part is known as ora serrata, from where the retina begins. From where the retina begins, this part is known as ora serrata. Okay, right? So coming to the notes here, the nervous coat of the eyeball, the nervous coat of the eyeball, that is known as neural or inner layer. It has your neural layer, which is known as retina. So why it is called neural layer? Because it is the one which senses the light. Okay. Retina has two parts, as you can see here. Retinal pigment epithelium, that is the epithelial part, and neurosensory retina, the neurosensory part which senses the light. Okay. And the neurosensory part contains the photoreceptors. The neurosensory part contains the photoreceptors. Then we have optic nerve. Optic nerve, how the optic nerve is formed? Optic nerve is formed by the axons of the ganglion cells, which are again present in the, it is the part of the retinal layer. The ganglion cells are also present in the retinal layer. Optic nerve is formed by the axons of these ganglion cells. Axon means actually the nerve fibers. So that is why we call it as neurosensory, neural. It's kind of a neural tissue. Retina has multiple layers. We'll be discussing later on. And the axons of the ganglion cell layer, they form the optic nerve. Okay. They form the optic nerve. So this is your third layer. Okay. So I hope you have a fair idea of the eyeball anatomy now, so that we, in the figures, in the diseases, you will see this cut section there. So now you can make out that how the different parts of the eyeball, which we use, how they are structure wise. Okay. So just a revision here. This figure is just for your revision here. So what are you seeing here, beta? First of all, the outer coat. The outer coat made up of cornea and sclera. Okay, just go with me. The outer coat made up of cornea and sclera. Okay, the sclera is covered by a layer called conjunctiva. Okay, sclera in front is covered by a layer called conjunctiva. Okay, and this junction here is limbus. This junction here is limbus, junction between cornea and sclera. Okay. Then comes your middle layer. Then comes your middle layer. So in the middle layer, in the front is iris. This is iris. Then there is ciliary body. Okay. So as you see, this is ciliary body, smooth and the processes. And then behind is your choroid. This is your choroid. Okay. Iris, ciliary body, choroid. And then innermost layer is retina. Innermost layer is retina. And optic nerve also arises from the posterior part, from the nerve fibers of ganglion cells. Ganglion cells. Okay. So this is how these coats are there. This is how these coats are there. Okay. Now, as I told you, it's a ball. So inner is a cavity. Inner is a cavity. Now, what is there in that cavity? That is called the chambers or the segments of the eyeball. Inner part of the eyeball is divided into two segments by lens. Now, what is this? This is your lens of the eyeball. This is your lens of the eyeball. And as you can see, the lens of the eyeball is dividing the eye into a posterior segment and anterior segment. Okay. It is dividing the eye. It is dividing the eye into a posterior segment and an anterior segment. Okay. Right. So now let's go on to the segments and chambers of the eyeball. The inner part, the segments and chambers of the eyeball. So eyeball is divided into, you can write here, two segments. Eyeball is divided into two segments. The part which is posterior to the lens, the part which is posterior to the lens, that is behind the lens, is known as posterior segment. The part which is behind the lens is called posterior segment. It is also known as vitreous chamber. Okay, it is also known as vitreous chamber. Okay, so where it is? This part, beta. Behind the lens. This behind the lens is your vitreous chamber. Vitreous chamber. Okay, right? And the part which is in front of the lens, the part which is in front of the lens, including it, that is known as anterior segment. Okay, so part of the eyeball which is in front of the lens, including the lens itself, is called anterior segment. What is behind the lens is posterior segment, which is front of the lens along with the lens is known as anterior segment. Okay, so this picture, this figure is now here showing you how the anterior segment. Okay, this is your lens, beta. This hole is your lens. Lens is suspended in the eyeball. Okay, the part which is behind, so it means this part is posterior segment, but this whole part, including the lens, from lens to cornea, this is anterior segment. Got an idea, beta? This is anterior segment. The whole part is the anterior segment. Now this anterior segment, look carefully. Now this anterior segment has two chambers. The anterior segment has two chambers. One is known as anterior chamber. Anterior segment has two chambers. One is known as anterior chamber from iris to cornea. I will make you write down so that you remember also. So first of all, see here, Anterior segment has two chambers. First is anterior chamber from iris to cornea. And this part is known as posterior chamber. Here, let me mark it with a different color here. This part is known as posterior chamber. Okay. This is anterior. So anterior segment is divided by iris into anterior chamber and posterior chamber. Right? You can write here. Please write down. The anterior segment has an anterior chamber. The anterior chamber is known as, friends, AC. So anterior chamber is known as AC. It is between cornea and iris. So you can write it like this. The part which is between cornea and iris is known as anterior chamber. And the second part is posterior chamber. Posterior chamber, which is known as PC, not Priyanka Chopra. Posterior chamber, PC, which is between iris and lens. So have this idea, beta, how these anterior segment is divided into two chambers by iris. Between cornea and iris, we call it as anterior chamber. Between cornea and iris, we call it as anterior chamber. And the small part between lens and iris, we call it as posterior chamber, AC and PC. Okay, right? Okay, now going ahead, what is there in these chambers? Now these cavities are not empty, otherwise the eye will collapse. There has to be some material in these cavities. So these cavities are filled by two kind of material or fluid, you can say. What? First is your aqueous humor. Now, aqueous humor, please write down. It is a fluid filling anterior chamber, AC, and PC. So both anterior chamber and posterior chamber, they are filled by a fluid which is known as aqueous humor. And I told you from where this aqueous humor comes from. It is secreted by ciliary processes of pars placaita. This can be your question, beta. This can be your question, friends, that aqueous humor, or what is the function of the ciliary processes of pars placaita? Pars placaita was a part of ciliary body, which is the middle layer. So aqueous humor is secreted by ciliary processes of pars placaita. Aqueous humor is secreted by ciliary processes of pars placaita. It is a fluid which fills anterior chamber and posterior chamber. Okay. And what is the function of this aqueous humor? It is not just like a waste fluid. It provides nutrition to your cornea and lens. Because both cornea and lens, they are avascular structures. They do not have blood vessels. Cornea and lens, they are the avascular structures of the eye, but they have to get nutrition. They don't have a blood supply, but they have to get a nutrition. So this nutrition is provided by a fluid which is called aqueous humor. Okay, friends? Right. Now, aqueous humor is filling anterior chamber and posterior chamber. Now, what is there in that posterior segment or the posterior the vitreous chamber, the posterior segment. Now it is filled by vitreous humor. It is filled by vitreous humor. Remember, come back to the figure if you have forgotten what is the vitreous chamber. The whole part, this whole part which is behind the lens, this whole part, this whole cavity which is behind the lens, this vitreous chamber, it is filled by, if I like fill it like this whole part is filled by what? Vitreous gel. Vitreous gel. Okay. So coming back here, come back here, beta, vitreous humor. Now this fluid is known as vitreous humor, or also known as vitreous gel. So there is a large cavity there. The volume of the vitreous gel is about 4 ml. What is the function of the vitreous gel also? It provides nutrition. Again, it provides nutrition to retina and lens. Mainly lens. It provides nutrition to retina and lens. It also maintains the intraocular pressure, because otherwise the eye will collapse. If there is no gel, if there is no fluid in the eyeball, the eye will collapse. So it maintains the intraocular pressure, the vitreous gel. And from where the vitreous gel comes, the question comes to mind, that from where the aqueous humor is produced by ciliary processes, but from where the vitreous gel comes. The vitreous gel is formed during embryonic development. It's a gel-like. It is formed during embryonic development. It is not secreted. It is formed during the embryonic development, and it stays like that. Okay. So got an idea, beta? This I have spent a bit more time on the anatomy and telling you about the different structures, so that you have a fair idea what is there in the eyeball. Only then you will be able to understand the diseases. Okay. So a very quick revision. Eye is like a ball. It has outer coats. I have already told you: fibrous coat, vascular coat, neural coat. Then within this eyeball, there is a lens in the center, which is suspended in the eyeball. Okay. Now this lens divides the eyeball cavity into two parts. Behind the lens is the posterior segment. In front of the lens is the anterior segment, including the lens. Okay. Now this posterior segment is also known as vitreous chamber. It is a large cavity, 4 ml, nearly 4 ml. Okay. Now this posterior segment is filled by a gel-like fluid, which is called vitreous humor, and which is formed during the embryonic development. The anterior segment, the anterior cavity, is further divided into two parts: anterior chamber, AC, and posterior chamber, PC, by the iris. Between iris and cornea is anterior chamber. Between iris and lens is posterior chamber. Okay. And these two cavities, anterior chamber and posterior chamber, they are filled by a fluid which is known as aqueous humor. So they are filled by the fluid which is known as aqueous humor, which is secreted by the ciliary processes. So this whole is about anatomy and how these different structures and segments are there. Okay. So I hope it is clear to everyone. Now let's move further. Let's move further. Some important things about the anatomy. Now, how the eyeball gets its blood supply with it. Come to your notes, back to your notes. How the eyeball gets its blood supply. The blood supply of the eyeball is by ophthalmic artery. By ophthalmic artery. Please write down: ophthalmic artery is a branch of internal carotid artery. It is a branch of internal carotid artery. Now, ophthalmic artery gives two kind of branches. One is your central retinal artery. One is your central retinal artery, which supplies the inner retinal layers. Central retinal artery, which supplies the inner retinal layers. Okay. And then there are ciliary branches, or what we call as ciliary arteries. Now, ciliary arteries, there are three types of the ciliary arteries. Okay. Long posterior ciliary arteries. Actually, it is not a single one. You can make a correction here. They are the multiple small branches. So one is your long posterior ciliary arteries. Now these long posterior ciliary arteries, they pass through the space above the choroid. They pass through the space above the choroid, that is suprachoroidal space, and they supply anterior segment. Okay. So long posterior ciliary arteries, they supply anterior segment. They are arising from behind, but they come forward. That is why they are long, and they supply anterior segment of the eye. Okay. Then short posterior ciliary artery, short. So they remain behind only, and they supply the choroid layer. Short posterior ciliary arteries, they supply the choroid layer. And then there is a group of anterior ciliary arteries which supply the mainly extraocular muscles. They supply mainly your extraocular muscles. Okay, so these are the different ciliary branches. Long posterior ciliary, which come forward and supply the anterior segment. Short posterior ciliary, which supply the choroid, and anterior ciliary arteries, which supply the extraocular muscles. So this is about the blood supply of the eyeball. Okay. Then coming to the nerve supply, please write down the note here. Nerve supply. Now the sensory supply to the eye is through fifth cranial nerve. The sensory supply of the eye is through fifth cranial nerve. And which is the fifth cranial nerve? Your trigeminal nerve. So the sensations which we feel, like, they are through the trigeminal nerve, fifth cranial nerve. Okay, right? So this can be sometimes asked as a question. The sensory supply is the fifth cranial nerve or trigeminal. Okay. Now coming to some important points in the embryology. Now in embryology, beta, this figure is just to tell you how the kind of the structure of the eyeball develops during embryology. We just need to know few important points. I will just explain a bit here. No need to write that because that is really not needed for exam. What is needed for exam, I'll make you write. Okay. So just listen to me here and looking at the figure. Now in the embryology, how the eye starts developing, it starts developing as an optical vesicle. Okay. The neuroectoderm of the prosencephalon. Okay. The neuroectoderm makes an optical vesicle. Then the surface ectoderm, which is close to that area, that starts forming the lens. Okay. Now what happens? This lens kind of invaginates. It invaginates the optic vesicle, making an optic cup. This is just for understanding. Actually, this is not needed for your exam. So it starts as an optic vesicle. Then the lens invaginates that vesicle, making a optic cup. Okay. Then gradually it starts invaginating it further, and lens becomes enclosed in the optic cup. Lens separates from the surface ectoderm and it becomes enclosed in the optic cup. Then this is optic stalk, where it is will be forming ultimately the optic nerve. Then different kind of mesenchymal tissue around this will form the other structures, the mesenchymal layers. And gradually we get the shape of the eye, the cornea develops, sclera develops, and the eyeball gets its shape there. Okay, so this is just a basic understanding. It is absolutely not the detailed one because that is not needed. So it starts a kind of optic vesicle, converts into optic cup. The lens gets enclosed within this optic cup. There is formation of the mesenchymal, different structures from the mesenchymal layer, and ultimately we get the whole shape of the eyeball. Okay, so the points which we need to remember, now let's write down those, the points which are needed for the embryology. Please write it down. So the important structure, so there will be three main layers, the embryonic layers from which the different structures will be developing. Okay, your surface ectoderm, neuroectoderm, and the mesenchymal layer. Okay, now what happens, please write down, from surface ectoderm, the structures which, important structure which develops is your lens, because this can be asked as a question. Okay, from the surface ectoderm, the important structure which develops is your lens. Then from neuroectoderm, the important structures which develop, that is your retina, optic nerve, and vitreous gel. Okay, again, this can be your theoretical question. From neuroectoderm, the important structures which develop, please write down, they are retina, optic nerve, and vitreous gel. Retina, optic nerve, and vitreous gel. Okay, and in the last, from mesenchymal layer, the mesenchymal tissue, from mesenchyme, the important structures which develop, they are your sclera, choroid, cornea, except epithelium. Cornea has different layers. So except the epithelial layer, rest of the cornea, and also extraocular muscles. So these points are, I'm making you write from exam point of view, from where the question can come. Okay, so from mesenchyme, the important structures which develop, they are your sclera, choroid, cornea except epithelium, and extraocular muscles. Okay, so these are the only points which you need for your exam from embryology. Right? I hope everyone has written. Please write it down. Surface ectoderm, lens, neuroectoderm, retina, optic nerve, and vitreous gel. From mesenchymal tissue, sclera, choroid layer, cornea except epithelium, and extraocular muscles. Okay, so this is about embryology, and something about eye at birth. Okay, so we have studied now the gross anatomy, gross sizes and everything. Now there are certain peculiarities. The eyeball is not exactly same as an adult eyeball when a child is born. When a child is born, the axial length is smaller. The eyes grows afterwards. Eyeball grows afterwards. So I told you what is the average axial length, anterior-posterior in the beginning, 24 millimeter. So adult eyeball is 24 millimeter. But at birth, it is a small eye. The axial length is 16.5 millimeter. And we know the small eye is hypermetropic. The small eye will be having a refractive error of hypermetropia. Okay. How much hypermetropia? It is normally in the range of +2 to +3 diopter. This is a normal physiological refractive error in a child when it is born. Okay, so every child is born hypermetropic because the axial length is small. How much hypermetropia? Because this can be your question. How much hypermetropia? +2 to +3 diopter. The lens of the eye in a child, the lens of the eye in a child is spherical in shape. How it is different from adult? In adults, we call the lens is biconvex. So this is the shape in adults. This is the shape of the lens in adults. It is called biconvex. But in children, it will be spherical. Children means at birth. So at birth, it is spherical like this. Okay. Then macula. So macula, what is the macula? It is part of the central retina. So I told you the inner layer, the retina, the central retina is called macula. It is undifferentiated. Undifferentiated means it is not fully mature. It is immature. Okay? So these can be your questions. Okay? That eyeball at birth is smaller in size. Because of the small size, a newborn baby will be hypermetropic, which is known, and about how much hypermetropia? +2 to +3 diopter. The shape of the lens is spherical, not a biconvex like adults. And the central retina is immature. It is not fully developed when at birth, undifferentiated. It is not fully developed. Okay, so these are the differences, main differences, which we need to know about the eye at birth and an adult. Okay. So this summarizes your basic anatomy of the eye. This summarizes your basic anatomy of the eye. I hope you have got a fair idea how the different structures of the eye and different chambers and all that stuff is there. So these may be very, very new terms for you. Don't kind of worry. You will be like, becoming familiar to all these words as we move further. Okay, because we'll be doing the detailed anatomy and you will be knowing these words again and again: retina, choroid, iris, vitreous. So all these terms, they may be very confusing at the moment, but the more you learn, the more you read, you'll be able to understand them better. Okay, friends, so can we go ahead and move on to the physiology of the eye, that how the different things are happening in the eye? Okay, and I'll give you a tea break after that once we do some more. Okay, so let's move on to the second chapter. Let's move on to the second chapter, beta, physiology of the vision. Friends, physiology of the vision. Because this whole eye is for what? The purpose of the eye is to see. Now let's understand how do we see, how the different parts of the eye, they help us see. Physiology of the vision. So, the light has got a lot of spectrum. There is ultraviolet light, there is infrared light, there is visible light. We are not able to see all the wavelengths of the light. What we are able to see is the visible spectrum. So what we are able to see is your visible spectrum, and in the visible spectrum, the wavelength of the light which is visible to our eye is 380 to 750 nanometer. Less than 380 will be your ultraviolet light. We cannot see the ultraviolet light. More than 750 is your infrared light. So infrared light we are not able to see. Human eye can see only between 380 to 750 nanometer, which is the visible spectrum of the eye. Now, how do we actually see the eye, the light? Okay, or how do we actually, what is the process, when the light comes to our eye, we are able to make an image. Okay, so there are three steps. There are three steps involved in formation of image. Okay, so the three steps in image formation are, the three steps in image formation are, so supposedly this is a pen. How do I see this pen? So first of all, there should be visible light there. If I place this pen where there is no light, I will not be able to see. Okay, so the first of all, the light has to fall on this pen. Now, every object on which the light fall, it reflects the light. Okay, please understand better. Every object to be seen, the light has to fall on that object, and then that light will be reflected from that object. Now, our eye will receive that reflected light. Now, this light is received by who senses the light in the eye? Which structure senses the light in the eye? They are photoreceptors of retina. Photoreceptors. Sensing the light. The photoreceptors, they sense the light. So the light which is coming from this pen, reflected light, will be sensed by photoreceptors. Now, the second thing these photoreceptors, they do, they convert this light signal into neural signal. They convert the light signal into a neural signal. Okay, and this process is known as phototransduction. Photo means light, transduction means the energy form is changed. From light signal, it is now converted into neural signal. So the first step in the image formation is that phototransduction, conversion of light signal which has been sensed by photoreceptors into neural signal. Okay, now this neural signal has to be passed. Okay, I cannot process the image. This signal has to be passed to the brain. So how does it goes? The transmission, the second step is the transmission of the signal. The second step is transmission of the signal. The transmission of the signal occurs through different layers of the retina and optic nerve. Okay, so this occurs through layers of retina. Especially two cells are very important to know here. Write down the name. The bipolar cells and ganglion cells. These are the cells which are present in retina. So they help in transmitting this signal. Bipolar cells and ganglion cells. Okay. So keep writing what I'm writing, because this is the addition. So I hope you are writing along with me. Bipolar cells, friends, and the ganglion cells, these are present in retina. They transmit the signal, and then signal passes through your optic nerve. Optic nerve, which is your second cranial nerve. So from retina, the signal passes to optic nerve. And optic nerve goes into the brain. It becomes a path of the visual pathway. And then the third, ultimately the signal reaches visual cortex, which is in the occipital lobe of the brain. where it is perceived or processed. So perception, the actual image perception is by the brain. Eye, the purpose of the eye is to receive the light from the object, convert it into the signal, the image signal, but the processing occurs in brain. So actually it is your brain which sees. Okay, so these are the 3 steps of the image formation. Phototransduction, which occurs by the photoreceptors, the transmission of the signal, which is by the retina and optic nerve, and visual perception, which is by the visual cortex in the occipital lobe. Okay, so this is how we are seeing. Now, going ahead, so this is just to let you know about retina. We'll be discussing more in the retina. This is just to give you an idea what I just told you. Okay, so retina is a multi-layered structure. Okay? So this layer, the layer of rods and cones, there are 2 types of the photoreceptors. Okay? Rods and cones, we'll be writing it. So these are the one which sense the light. And then this signal from this is passed through the different layers of the retina. From the different layers of the retina, it goes into the nerve fiber. Okay? And this nerve fiber layer ultimately forms the optic nerve. So this is how the light signal is passing. This is just for understanding how the light signal is passing from the photoreceptors through the different layers of retina and ultimately going to the optic nerve. Right? So that is why we call retina as the neural layer. We call retina as the neural layer because it has these nerve cells. It has these nerve cells. Okay. So this is about about the retina. Don't even bother if you don't remember at this point of time. Retina has an epithelial layer, which is called RPE, and a neurosensory part, which is multi-layered, 9 layers. And it is the neurosensory part which is helping us transmit this nerve signal. Okay, this is just for understanding better. Don't get confused about different things, for understanding how the nerve signal is getting passed. Okay, the thing to remember here is about your photoreceptors. There are 2 types of photoreceptors. Write down 2 types of photoreceptors, which I have written here. One is your rods. One is called as rods. The other are known as cones. Okay. So there is different function of both. The rod photoreceptor, rods. The rods photoreceptor, please write down. They are for scotopic vision. Scotopic vision. What do I mean by scotopic vision? In dim light conditions. Dim light, or even you can say night time. Okay. When the light is low, dim light or night time, that is known as scotopic vision, and it is the rod photoreceptors which help us sense the light during dim light. Rods. The second are your cones. Cones are responsible for, please write down, photopic vision. They are responsible for photopic vision. Photopic vision means the vision during bright light. For example, in a bright sunlight. Okay, so this can be asked as a question. Rods are responsible for scotopic vision. Cones are responsible for photopic vision. What is the difference? Scotopic means dim light vision. Photopic means when you see in a bright light. Both rods and cones are two types of photoreceptors. They are two types of photoreceptors. Okay, right? Now, what is the chemical basis of this visual process? What these photoreceptors are actually doing? You know, I told you they are sensing the light, but what is actually happening? The important thing to remember here is these photoreceptors, they have visual pigments. They have got a chemical which is called visual pigment. This visual pigment of the rods is known as rhodopsin, and in cone it is known as iodopsin. These photoreceptors, they contain visual pigment. The visual pigment of rods is known as rhodopsin. Cones is known as iodopsin. Okay. Now, what happens when the light strikes? Now, when light strikes, okay, please, you can write down in the space here. When light strikes on these photoreceptors, these visual pigments, they break down. These visual pigments, they break down. And when they break down, then action potential is generated. When they break down, then action potential is generated, which helps in now making a neural signal. Okay, so you can call it as the chemical process of the, like, you know, what is happening basically. The light is sensed by photoreceptors. These photoreceptors, they have pigment, rhodopsin and iodopsin. This pigment will break down. So once the pigment breakdowns, along with the action potential is generated, because retina is a neural tissue. So now this light signal gets converted into the neural signal, which is then passes from optic nerve to brain, as I told you. Okay? So you have got a concept now that how the light gets converted into a neural signal. And this neural signal, let's go forward. This neural signal then passes through this visual pathway. This neural signal has to go from eye to brain. So it passes through this visual pathway, what we call as visual system. Okay? This whole is your visual pathway. So at this point of time, just try to kind of just familiarize yourself to this figure. Here we are not going to remember this. We'll be discussing visual pathway in detail. This is just to let you know. Okay. So this is your eye. This inner layer is your retina. So ultimately the light has been sensed by retina. Then it passes to the optic nerve. This is your optic nerve. It passes through the optic nerve. And then it passes through different parts of the visual pathway. These parts are, we call them as optic chiasma, optic tract, lateral geniculate body, optic radiation, and then the striate area or visual cortex. So don't bother if you don't remember all these things at the moment. This is just to let you know from retina to brain, what is the visual pathway. From retina to brain, what is the visual pathway? Retina to brain. Okay. And the area of the visual cortex is known as area number 17. So this sometimes can be your theoretical question. This can be your theoretical question. The area number 17 is your primary visual cortex. Primary visual cortex. So got an idea, friends? The light passes through different structures of the eye, goes to retina, sends in your brain, where primary visual cortex, which is area number 70. Okay. Okay, so now we are going to go little more forward there. Now we are going to go little more forward there. Now come to the clinical aspect. Now we are going to come to, so we have done the basic theory, we have done the anatomy, we have done the theory of the vision. Now let's go to the clinical thing, which will be more interesting for you. So when I say clinical thing means how I am going to see whether a person is having a good vision, how a person is having a normal vision. Because if I have to understand the eye diseases, the vision is the main thing. If there is any problem in vision, it means there is some problem in the eye. So the different eye diseases, they can cause vision problem. So I have to understand now how I can examine the visual status of a person. Okay. Now, if I have to do the test for vision, now vision is not a single thing. It is not a simple, you can say like, just that whether I am able to see or not. There is quantity and there is quality of the vision. Okay. So we call it as 4 components of the vision. We call it as 4 components of the vision. When you see at an object, okay. So for example, when you see at this phone or anything, or when you read, what are you seeing? First of all, you are able to see whether there is light or not. Okay. In the 4 components, first of all, what you see is, there has to be light sense. If you cannot sense the light, you cannot see. So the basic thing is the light sense. Then you know what is the shape or form of this object, that whether it is a square or it is a symbol or it is, you can see the shape, that is form sense. Then third is your color sense. You can see whether it is black, red, white, blue, whatever, color sense. And the fourth, we call it as contrast sensitivity. Contrast means whether it is bright in comparison to the background or it is light in comparison to the background, contrast of a thing. Okay, so these are known as 4 components of your vision. So your vision is remarkable. Like, eye is like, we call it as a camera. High resolution cameras. We are so mad about our phones, smartphones, that the camera resolution should be best. So there is nothing better than the resolution of your eye. Eye is the best God-made camera. And that is what we are like, trying to achieve in all these smartphones and all that. And this vision has 4 components. What are these 4 components, as I told you? So first is your light sense, that whether you are able to perceive light or not. That is the first component. If you are not able to perceive light, it means you have no vision. So the perception of the light, perceiving the light is the basic requirement to see. Okay. Then you have form sense, how a thing looks like. And that is the basis of your visual acuity. Okay. Visual acuity means, like, when we measure, the visual acuity is measurement of form sense. When you read, it is called your visual acuity, how good you are able to read, whether you can read small letter or you can read a big letter. That is the measurement of your form sense. And then color sense, you are able to see the different colors. As you know, people can be color blind. So a normal color sense. And then 4th component is contrast sensitivity. So I will show you how the contrast sensitivity is. I'll show you the chart. You'll be able to better understand. So when we are testing a vision for the person, we have to see that all these components, they should be normal. Only then a person has normal vision, friends. Okay. Only then a person has normal vision. So in the coming part, we are now going to learn how to do different tests of vision. Okay? So first of all, let's see how to test the visual acuity of a person. Now, there is a concept what is called as minimal angle of resolution, MAR. Okay, now this is for your understanding, beta. Minimum angle of resolution, MAR. You understand resolution. Better resolution means better visual acuity. Camera which has better resolution gives you sharper pictures, isn't it? So if the resolution is good, only then you are able to see it more clearly. So there has to be some minimum resolution to see. This minimum resolution to see is known as minimum angle of resolution. Okay. So if there is there is an object, supposedly there is this kind of an object. Now this object, and this is your eye. Okay, this is your eye. This is for understanding, even if you don't make this picture. Now, the different parts of the object, the light will be going from the different parts of the object. Now, this image will be seen sharply if it forms a minimum angle between 2 points. There has to be minimum angle which is made at the eye so that we can see that object. That is known as minimum angle of resolution. And this minimum angle of resolution is 1 minute of arc. Okay. So degree, we know angle is called in degree, but when the angle is very, very small, then it is measured in minutes of arc. Minute of arc is the measure of angle. Just like angle degree, 1 degree, 2 degree. But when it is very, very small, then we call it as, then we measure in minute of arc. So minimum angle, which must be subtended by 2 points in space. These are 2 different points, 1 point, second point. There has to be at least 1 minute angle, 1 minute of arc angle. Only then we will be able to see 2 points differently. So if we are able to see 2 points differently, only then we will be able to see the shape of that object. Okay. So supposedly the shape of the object is like this. Now, whether it is like this or like this, it will be seen only if the minimal angle of resolution is met. Okay. So most of the students, sometimes they get confused about this minimum angle of resolution. Nothing to get confused. This is basically the minimum angle which should be there between the 2 points, so that I can see that as a separate point. Otherwise, everything will appear similar. The shape is seen if there is a minimum angle of resolution. Okay, so thing to remember for your exam is how much is the minimum angle of resolution. Okay, so for your exam, the thing to remember is minimum angle of resolution is 1 minute of arc. Only then we can see the object. 1 minute of arc. Okay, right. Now going ahead. Now this forms our basis of testing of the vision. Testing of the vision. Okay. Now a patient, now think of like, you are sitting in a clinic. Think that you are sitting in a clinic now. A person comes to you and you want to check the vision of that person. That whether this person has normal vision or the person does not have normal vision. Okay. Now, what are we going to do? So that is your vision testing. That is your vision testing. So we do 2 types of the vision testing. One is your distance vision. One is your near vision. So what we do for the distance vision testing. Okay, now get into the frame, friends, that you are an eye doctor or you are a doctor and you are sitting in your clinic and a person comes and you have to check vision for that person. So the distance vision testing is done by using this chart. This chart which you are seeing here, this is Snellen's chart. This is Snellen's chart. Please write down. This is the most commonly used chart. Most commonly used chart for testing the distance vision of a person. Testing the distance vision of a person. Snellen's chart. Now there can be image-based question also on Snellen chart, that they show you chart and identify. There can be theory question. The Snellen chart is used for distance vision testing. Now, what is there in this Snellen's chart? Okay, so first of all, I'm going to tell you, everything is written also below. So first try to concentrate on this chart. Now you see this chart has different letters. The top letter is biggest in size, then smaller size, smaller size, smaller size. And this last line is the smallest one. So this is the biggest one and then smallest one. Okay. Now a person is to read from this chart, starting from the top line. Okay, so starting from the top line and a person goes down, keep on reading T, V, H, X, U, A, H, T, Y, O, and so on. So how much you can read will give us your visual acuity. Okay? So whether you are able to read one line, two line, three line, or all the lines. So that will tell me what is your visual acuity. Okay, so this is how we measure the visual acuity of the person. Okay. Now, what I have written here, 6 by 60, 6 by 36, these are the measures of the visual acuity. Okay. So how this is done? So first of all, let's go down here. Let's learn something about Snellen chart. So how do we test the visual acuity? You ask the patient to stand at 6 meter distance. Ask the patient, there is a chart on the wall, okay, and the patient is standing at 6 meter distance. If a person is able to read the last line at 6 meter distance, that is your 6 by 6 visual acuity, which is the normal visual acuity, right? So which is the 6 by 6 line? This second last actually. This line here represents actually 6 by 5. So you ask the person is sitting at 6 meter distance. If a person is able to read this line from 6 meter distance, then the visual acuity is 6 by 6. 6 by 6. Okay. Now, what does the 6 by 6 means? The 6 by 6 means 6 tells you, 6 by 6 means this denominator is the distance from which a normal person can read this letter. Okay. And numerator is from where the patient is reading. Okay, so please listen once more. 6 by 6 means the denominator, it's written also here. Denominator is distance at which a normal person can read that letter. And numerator is at which the patient is reading. So what do I mean by this? So supposedly this top line, this top letter, here the vision is called 6 by 60, means a normal person can read this at 60 meter also. The chart is there. The letter is so big that a normal person can read at 60 meter. But the patient is reading at 6 meter because patient has poor vision. Patient has to come close to the chart. So if the person is able to read only top letter on the chart, we call the vision is 6 by 60. because a normal person can identify this letter from 60 meter, but this patient is able to see at 6 meter. Okay, so we call this vision as 6 by 60. Now, if the person is able to read this third line, this is called 6 by 24. This is just for understanding, beta. Okay, 6 by 24. 6 by 24 means a normal person is able to read this at 24 meter. This line should be visible to a normal person at 24 meter. But patient is able to see only at 6 meter. So patient has to come closer. Right? So 6 by 24 vision is better than 660 vision, but less than the normal vision. You got my idea here? Okay. So if the person is able to see bigger letters, the vision is poor. If you are able to see the smaller letters, the vision is good. Okay. So 6 by, whether the person is able to see 6 by 60, 6 by 36, 6 by 24, all these are the measurement of how much the person can see. Okay, so got it, Beta? So normal vision is considered to be at least 6 by 6. Normal vision is considered to be at least 6 by 6. So this is the normal vision, how much a person should be reading. Now, supposedly the vision is so poor that patient is not able to read first line even at 6 meter distance. The vision is so poor, even the top letter is not visible. Then you bring the patient more close to the chart to measure the vision. You bring him more close to the chart to measure the vision. By 1 meter, you ask the person to read at 5 meter distance. Then the vision will be called as 5 by 60 because now the distance is 5 meter. Okay. Then you, if not even at that, you bring him more close, 4 meter. If the person is able to see at 4 meter the top letter, the vision is 4 by 60. So you can bring him more close, 3 by 60, 2 by 60, 1 by 60. But even if the patient is standing at 1 meter from the chart and not able to see the top letter, it means the vision is very, very poor. It means the vision is very, very poor. Then what do you do? How can you, we have to measure the vision. You have to have some quantification of that. So even if it is not visible at 1 meter distance, then you ask the person to count to your finger. Okay. We call it as counting finger vision. You show your fingers, whether these are 4 finger, 2 finger, 1 finger, whether patient is not able to see anything on the chart, you ask him to count your finger. That is called counting finger vision. If vision is so poor, patient is not even able to count, then you do what is called as hand motions. You do hand motion. You wave your hand like this. Wave your hand like this. That whether patient is able to see any hand motions. Okay. And if the vision is even, person is not even able to see your hand, then we do perception of the light. That is, you shine torch light. You shine torch light and ask the person whether he is able to see the light or not. That is known as perception of light. And you check projection of light rays, whether person is able to tell the direction of light. Whether the person is able to tell whether the light is coming from right side, left side, above, down, that is called projection of light. Okay, so this is how we measure the vision. This is how we measure the vision. Okay, so a quick revision how we measure the vision. So for the vision measurement, the chart which is used most commonly is Snellen chart. And what is there in Snellen chart? It has letters of different sizes. The top letter is big, then small, and smallest. The person will initially stand at 6 meters and read from the chart. If the person is able to read the 6 by 6 line, that is the normal vision. If the patient is not able to read that line, then above, whichever line he is able to read, you can mark that as the vision. Whether the vision is 624 or whether the vision is 660 and so on. Okay. If the person is not able to read any letter at 6 meters, then you bring the person close to the chart. Okay, you ask the person to read from 5 meters, from 4 meters, and so on. And you note the vision of that person. If the person is not even able to see at 1 meter, you make him count your fingers. If the person cannot count the finger, you move your hand motions. If the patient vision is so poor that even the hand motions are not possible, you show him torch light, that if he can sense the light or not. And you ask him from which direction the light is coming. Okay? So that will help us know what is the vision of the person. Okay? So now today all of you have learned how to check the vision of a person. Okay? Now, something for the theory. Now, is the Snellen letter any random letter that you show any chart? Can you make any chart, write on that ABCD and ask the person to read and measure the vision? No. The Snellen chart has certain specifications. Okay. The Snellen chart has certain specification. Now, each letter of the Snellen chart, it is a standard chart, beta. It is a standardized chart. Each Snellen letter has 5 segments. Okay, so if I take the example of letter E, okay, look at the figure here. If I take the example of letter E, so here you can see it has 5 segments. 1, 2, 3, 4, 5. Equal segments. And each segment has to subtend a minimum angle of resolution. So here comes your minimum angle of resolution. Each segment has to subtend minimum angle of resolution so that I can see that. Otherwise, I will not be able to differentiate different segments. To identify the letter, to identify the letter, each segment subtends an angle of minimum 1 minute of arc. Okay, so and each letter has 5 segments. So the whole letter, so this is 1 minute of arc, the whole letter should subtend 5 minutes of arc. Okay, so these letters are standardized. These are not some random letters. Each letter has 5 segments. Each segment should subtend minimum angle of resolution, which is 1 minute of arc. And the whole letter should subtend 5 minutes of arc. So why I am telling you this? This can also come as a question. Okay? So it is not some random letter. They can ask you about the Snellen letter. Each segment of the Snellen letter subtends an angle of 1 minute of arc, and the whole letter subtends an angle of 5 minutes of arc, and it goes according to our minimum angle of resolution. Only then we will be able to see the shape of that letter. Okay, right? Okay. Now, somebody Now this Snellen chart, the question should arise in your mind. It has English letters, A, B, C, D, English alphabet, isn't it? Now, if somebody does not know English, are there other language charts also? Yes, there are other language chart also available, but each letter has to follow the standard of the Snellen chart. Okay? Now, if somebody does not know any language letter, a person who is illiterate, how would you measure the visual acuity of an illiterate person who cannot identify English letter, Hindi letter, Punjabi letter, Urdu letter, whatever? So what do you do for that person? How would you measure the visual acuity, friends? So for them, we have other type of chart, come down here, what are known as Landolt charts. The person who cannot identify, or you can say illiterate person, any alphabet there, we use Landolt chart. Okay, so write down, these charts are used for illiterate person. The Landolt charts are used for illiterate persons or even for young children who do not identify the letters. So this can be your question. Landolt charts are used for illiterate persons or for the young children who cannot identify alphabets. So there are two types of the chart, as you can see here. One is known as C chart. One is known as E chart. Okay. So here you don't have to see, like, you don't have to tell that this is the letter. You have to tell which side the cut is there. Like the person will tell the cut is on the right side. The person will tell the cut is on the left side. The person will tell the cut is on the downside. Here they will see where these E is pointing out. It is pointing on the right side or it is pointing on the downside. So that way a person can tell you. Okay. So these C and E chart, this can come as your question. Landolt chart, which are used to test visual acuity for illiterate people or for young children who cannot identify the alphabets. Okay, right. Now, the second question should come in your mind is, are there any other charts? Yes. So there are few more charts which you should know from your exam point of view. Now, this is another chart which is known as ETDRS chart. ETDRS chart. Okay. Now, what is the full form of ETDRS? The full form of ETDRS is Early Treatment of Diabetic Retinopathy Study. Okay. Now, this can also come as the question. This had come as a question. What is the full form of ETDRS? You should know that Early Treatment of Diabetic Retinopathy Study. Now, how this chart is different from the Snellen chart. So here you can see every line has 5 letters. Okay, every line has 5 letters. So you can identify this chart if it comes as an image-based question. So please keep marking on your workbook where I tell you to write the question or to mark as image-based question, because then it can come as an image-based question. Okay, so when you revise later on, then you can see these high yield points, friends. So please keep marking along with me. Okay? This can come as an image-based question. Identify the chart. It is your ETDRS chart. And how I am going to identify it? This chart has 5 alphabets or 5 letters in each line. Okay, right? Now, so each line has same number of letters. And here, the testing distance in Snellen, the testing distance was 6 meters. In Snellen, the testing distance was 6 meters. And here the testing distance is 4 meters. So again, this can be your question. That what is the testing distance in ETDRS chart? 4 meters. And where do we use it? we use normally, this chart is used, please write down. This is used for clinical research studies. Okay. So when you are doing research studies, this is more standard chart. When you are doing clinical research studies, then you use ETDRS chart. Okay. Then you use ETDRS chart. So 3 questions for ETDRS chart. Number 1, what is the full form of ETDRS? Early treatment of diabetic retinopathy study. Second question, what is the testing distance? 4 meters. Where it is used? For clinical research studies. Okay, friends? Okay. So this was your for distance vision chart. Now coming to the near vision charts. So when we are testing the vision of a person, we have to check the distance vision as well as the near vision. So commonly used near vision chart are, there are 3 commonly used near vision chart. The names are written: Snellen near vision chart, Jagger near vision chart, Roman near vision chart. So they can ask you the name of the near vision charts: Snellen, Jagger, Roman. Okay. And how they look like, here is the image. So you see there are like, smaller, the bigger font, smaller, smallest. So font size is changing. And the person is reading it from near. The near vision is tested. Okay. So please write down. Near vision is tested at 33 centimeter distance. Okay. So a person will be holding this chart at 33 centimeter and reading. Okay. So there can be simple paper chart like this one, or these kind of illuminated charts. This is just to let you know that how these charts they look like. Okay. So these are the near vision charts. The near vision is tested at 33 centimeter, and these are the names of the chart. Okay, right? Okay. Now, regarding, before we take a break, just 5-10 minutes, then we will be having a tea break. So when a person, like, I'm talking about the vision. I told you so much about the vision. But is this vision, same from childhood or from birth to adult? No, the vision gradually develops. Okay, the vision gradually develops because we read when the baby is born, the eyeball is not fully formed. The eyeball is not fully mature. Similarly, our vision, the quality of the vision is also not fully developed. There are certain visual milestones from birth to the age when we acquire full vision maturity. Okay. So why it is important? Because the vision has to be tested according to the age, according to the extent of visual maturity. You cannot compare the vision of a 2-year child to an adult. The normal range is different because of the different milestones in the vision development. Okay, so this visual milestone, please concentrate here, please focus here. This is telling you what are the different ages at which different visual milestones they develop. Okay, so it's more of a theory. Let's learn the fixation. So first visual milestone is fixation. Fixation means how you are able to focus. So when a child is born, the eyes are wandering. The eyes are not able to focus. The focus or fixation starts at the age of 6 weeks and it is completed by 6 months. Okay, so I have given a 6, which will be easy to remember for you. Okay, this is a theoretical visual milestones. The fixation starts at 6 weeks of age and it gets complete maturity by 6 months of age. So then a child is able to focus. Okay, then the second milestone is known as fusion. What the fusion means? Beta, we have 2 eyes. Each eye is seeing separately. Okay, so we have 2 eyes. We have 2 hands. Both hands, they do work differently. Now, whether they are doing the same thing, when they do the same thing, then it will be fusion. Okay, both come together. See my 2 hands. 1, 2. Both come together. That is the fusion. So that is the quality of both eyes. Both eyes, they fuse. They fuse their image. Otherwise, you will see everything double. One eye, right eye is seeing one image, left eye is seeing one image. These 2 images are to be fused. This visual milestone is called fusion, so that we see object as single. This fusion develops by 6 months of age. Okay, so first of all, we have to focus. Second, we have to fuse. Then there should be stereopsis. Please correct the spelling here. S-T-E-R-E, stereopsis. The third quality is stereopsis. Stereopsis means we are able to see the 3-dimensional thing, isn't it? Not every animal is able to see 3-dimensional. Human eye can see 3-dimensional. That is called stereopsis. Okay. Now this stereopsis also, it starts developing at 6 months of age and completed by 6 years of age. So the different quality parameters, the milestone at different ages. Stereopsis completes by 6 years of age. And a child attains the adult visual acuity of 6/6 by 6 years of age. Okay, so 1 year child will have not the same vision. The second year child will not have the same vision. It is at the age of 6 years, a child attains complete visual development. Okay, so they can ask you this as a question. So this can be asked as the visual milestones can be asked as questions. So fixation, focus, fusion, fusion of 2 images, stereopsis, depth perception, 3-dimensional, and the visual acuity of 6/6, which is an adult visual acuity, is attained at the age of 6 years. Fine. So this you can remember for your exam. Now, the last thing before we go for tea break, how to test vision in children. How to test vision in children. So because I told you the vision is different in children, so the testing method should also be different. So how will you test vision in a child which is 6 months of age, for example? Or how will you test the vision in a child which is 2 years of age? So there are different methods. So the names of these methods are important to know. I will tell you briefly about what we do in this, but you don't really need to bother in the detail. The name you should remember. The name you should remember. Okay. So we divide the visual testing into 2 types of the test, the objective test and the subjective test. Objective test in which you observe, the examiner observe, and subjective means where the child will tell you. Okay, so objective test will be done for pre-verbal children. Pre-verbal means who cannot speak, Like a 6-month-old child cannot tell you whether he is able to see a car or toy. He cannot express. So we call them as pre-verbal children. For pre-verbal children, we will do objective test. And for older children, we can do subjective test. Now, the 3 tests which are done for the pre-verbal children, the name of the test are, first is Teller Preferential Acuity Test. The second is Visual Evoked Response Test. The third is Optokinetic Nystagmus Drum. Okay, so these are the 3 objective tests. 3 objective tests. So they can be asked as the question in your exam. What are the tests which you do for the pre-verbal children? So please remember the name: Teller Preferential Acuity Test, Visual Evoked Response, Optokinetic Nystagmus Drum. You have to remember the names. I'm going to explain a little bit about these tests. You don't really have to bother about, just for your concept, so that you are able to remember these names. Okay, so come forward here. What we do in preferential acuity, just concentrate on the figure, don't write anything. This is just to build the concept better. Preferential means preference. Okay, so here what you see, there is this child. He is being shown 2 kind of cards. This is a plain card. Okay, this is a plain card. And this is a card which has a pattern on that. These pattern, red and white, a black and white pattern is there. Now, if the child prefers the pattern, then it means the child has normal visual acuity. He can see the pattern, which is more attractive for a child. The pattern will be more attractive than the plain, isn't it? That is why we call preference. So you see whether the child is preferring any particular card. You see the eye where he is seeing. So Teller Preferential Acuity. If the child prefers a pattern, it means child is able to see better. Okay, so this is just for you to understand. No theory, no writing. Pattern card is preferred if the child has normal visual acuity. Okay. Now, another one I told you, optokinetic nystagmus. Now, optokinetic nystagmus means involuntary eyeball movement. Okay, nystagmus means involuntary eyeball movement. Here what we do, a child is there, a baby is there, you show him this drum, and you can see this drum also has black and white lines on that. Name of this drum is optokinetic drum, OKN drum. You rotate this drum in front of the child. Now, when we look at a rotating object, our eyes, they go on to a normal nystagmus, which is called physiological nystagmus, because we have to focus on a rotatory object. So the eye will start moving on its own. This nystagmus is called physiological nystagmus, which is normal. So if the child is able to see the rotating drum, you will notice the nystagmus in patient's eye. So it means the child is having normal vision. Okay, so it is again your objective test. You rotate the drum, you notice whether there is a appearance of any nystagmus or not in the child's eye. If there is appearance, it means the child has normal vision. Okay, right? And the third test which I told you in the pre-verbal children is your visual evoked response. Visual evoked response. In short form, it is also called VER. VER. It is just like your ECG or ERG. You check the electrical activity of the visual pathway. Just for your understanding better. You check the electrical activity of the visual pathway by placing electrodes. So these are the 3 tests for your exam. You have to remember what are the 3 tests for pre-verbal children: Teller preferential acuity test, visual evoked response, optokinetic nystagmus drum. The names. Okay, right? Then if the child is little older, that is age of 1 to 2 year, now he can differentiate between the different sizes. Okay, he can differentiate between the different sizes. So again, just remember the name of the test. So test which you do: Worth ivory ball test, Sheridian ball test, Boeck candy test, coin test. So here what are we doing? We are showing them the objects of different sizes. For example, whether the child is able to pick up the big ball, small balls. You can use the candies, you can use the coins. Okay, you tell the child to pick up a bigger coin or a bigger ball, and you can judge the visual acuity. Okay, so you should just remember the names. Okay, beta. Then if the child is little older, okay, please, you can do the correction here. Mark here 2 to 4 years. Please do the correction here, 2 to 4 years. Now, if the child is 2 to 4 year old, we can show them picture cards. We can show them picture cards. Okay. The 2 tests are based upon, you can show them the picture and they can match the picture, or they can tell whether it is horse, cake, train, or they can match the picture. The name of the test are important: Cardiff acuity test, Allen picture cards. Cardiff acuity test, Allen picture card. You show the picture, child can match with the card. If he doesn't speak, or he can even, because child can be shy also, or he can tell you what image it is. Please remember the name for your exam: Cardiff acuity test, Allen picture card. Okay. And in the last, if the child is now about, there is overlapping of the year age beta. Okay, so don't get confused. 2 to 4, 3 to 5, there is certain overlap also between the test. So if the child is around 3 to 5 years of the age, we use a chart which is known as Steiker. You need to remember the name, Steiker. The Steiker, the full form of Steiker is Screening Test for Young Children and Retarded, even the adults who are retarded. Again, it is a special type of picture test. So you can tell them to identify this picture. Steiker test. Just remember the name. And in the last, there is one more test for children. There is one more test for children, which is known as Sheridian HOTV chart. Okay. Again, this can be asked as a question. This is again a chart for children. So here you see only 4 letters are used. Not to confuse the child, we use only 4 letters. HOTV. So that is why it is called HOTV chart. Okay. Sheridian HOTV chart. Again, this is used for children between 3 to 5 years of age group. Okay. So here we take a tea break of 20 minutes before we join again. And you have got an idea. What we did just now, an overview. We learned about how to test the visual acuity in adults, different tests. We learned about the various visual milestones, and we learned about how to check the vision in children, different age group. Okay, so let's take a break of 20 minutes, and refresh yourself, have a cup of tea, have a cup of coffee, and we come back and join again. Okay? So let's break here. Welcome back again, friends. I hope you could have a cup of tea or coffee to recharge yourself. And we will be resuming from where we left. I hope all of you are back again, and let's resume. Now, these first few chapters are more of, as you are getting it, more of, making the basis. And I'm telling you the high yield points in between. From these chapters, from anatomy or physiology, most of the time you will get, as I told you, one-liner theoretical questions or some image-based questions, as I have told you. So I hope you have marked them, as I told you, along with highlighted them. It's difficult to, I got a query regarding how much percentage of question will be coming from each chapter. So it is difficult to say, because every exam, sometimes there will be more questions from one topic or from another topic. But as we go ahead, I will tell you there are certain topics from which certainly the questions come. So it's difficult to kind of distribute percentage wise, and we shouldn't be so rigid about that, whether 2 questions will come from this chapter. It's never like that. But yeah, there are some often repeated topics which we must know. I will keep telling you often repeated questions, expected questions, high yield points, so that when you are revising, then you focus on those points only, and you don't waste time on unnecessary things. Because for exam, the smart reading the high yield points and revising them, retaining them is very important, rather than spending extra time on extra things which may not be so high yield. So I fully know that, and I will guide you accordingly. Okay. So from these, the chapters which are regarding the basics, they will be more of one-liner questions, I have told you. So now resuming to the class once again, friends, I hope all of you are back here. So we were talking about, we are learning about the testing of the vision. Okay, we have learned about the distance vision testing, near vision testing, the testing in children. Now coming to the next topic, to check about contrast sensitivity. So as I had told you, contrast means how much is the brightness as compared to the background. So that is your quality of the contrast sensitivity. And to check the contrast sensitivity, the chart which is used is known as Pelly-Robson chart. Pelly-Robson chart, and this is how this chart looks like. So as you can see, the top line is having the maximum contrast, bright black in comparison to the white background. The lower one is little gray, then more light gray, light gray. So the contrast is decreasing as you are going from top to the bottom. So this will be the measure of your contrast. Whether a person is able to see the lowest contrast. So if you notice, the lowest line is also visible, the lowest contrast. Okay. So this will be the judge of your contrast sensitivity. This chart is known as Pelly-Robson chart. The question can come as one-liner and maybe image-based question. Okay. So image can come, you can identify the contrast, different contrast, name of the chart, Pelly-Robson chart, and it is used for measuring the contrast sensitivity. Chart for the contrast sensitivity. So mostly you can add here, the contrast sensitivity can be affected, especially various diseases. For example, if the patient is having cataract, if the patient is having glaucoma, if the patient is having optic nerve diseases. So the contrast sensitivity can go down. The vision may be normal. The reading on the Snellen chart may be normal, but the contrast sensitivity can go down. Right? So these are the diseases where we check for the contrast sensitivity. The name of the chart is Pelly-Robson chart. Then another feature of our vision is color vision. Color vision. So we can identify different colors. So testing of color vision is also one measure of testing your vision. Now in color vision, friends, there are three primary colors. Okay? Red, green, and blue are called three primary colors. And whole of our color vision is based upon the mixing of these colors. Now, this is known as trichromatic theory of color vision. Okay? So it is considered that our normal color vision is trichromatic, combination of three primary colors, trichromatic. And Young and Helmholtz, they gave a trichromatic theory of color vision. According to them, there are three types of the cones to identify three primary colors. So you have red cones, you have green cones, and you have blue cones. Okay, so three type of the cones for three type of the primary colors. So I told you in the beginning also, the cones are for the photopic vision, bright vision, as well as for your color vision. It is the cones which are responsible. Okay, so simple concept, the photoreceptors which are responsible for your color vision, they are your cones. And there are three types of the cones. Now, if there is deficiency or defect in either of these cones or in any of these cones, that will lead to defect in your color vision. Okay, so it is defect that will lead to defect of your color vision. Now, defect in the color vision can be partial or total. Okay? When it is partial color blindness or color defect, we call it as anomalous. Anomaly. Okay? Anomaly. It is not normal. It is anomalous. There is partial color blindness. So the partial color blindness is called anomalous. Or if a person is not able to identify any color completely, that means anopia, complete absence, anopia, that is total color blindness. Okay, so color blindness can be partial when we call it as anomalous, or complete, total, when it is called anopia. So these are the two terms, anopia, anomalous. Now, depending upon which color is defective. Okay, so if it is red color which is defective, if it is red color which is defective, I think I pressed some wrong button over there. I'm sorry for that. Let's resume. So we are talking about the color vision defect. Now, depending upon which color is defective, if it is the red which is defective, then it is called pro 10. Pro10 is for red. So if it is red defective, it is called protonamylase. If it is green defective, it is called deuteren. Deuteren is for green, deuteranamylase. If it is blue defective, it is called tritonamylase. Okay. So protan for red, deuteren for green, triten for blue. If it is partial, it will be enamylase. That is, a person is not able to see red. So red color is defective, protonamylase. Green is defective, deuteranamylase. Blue is defective, tritonamylase. And if it is complete blindness, then it will become anopia. If red is not seen completely, then we call it as protanopia. If blue is not seen completely, tritanopia. If green is not seen completely, deuteranopia. Okay, so simple thing, nothing difficult here. Partial means enamylase, anopia means complete. Now, if there is anopia, it means the patient no longer have trichromatic vision, because now one color is completely absent. So we call it as that now the patient has dichromatic vision. Okay, if it is anopia, it means the patient has dichromatic vision because one color is completely absent. Okay, right? Now, going ahead. The color deficiency can be congenital or acquired. Okay, a person can have congenital color deficiency. The congenital color deficiency, please write that down. It is mostly X-linked recessive trait. Congenital deficiency is mostly X-linked recessive trait. So it is seen in males. Congenital color deficiency will be seen in males. Okay? Whereas acquired is usually of two types. The blue-yellow defects are seen in retinal diseases. Now, this again can be a question. If a person is having a retina disease, which type of color deficiency is common? That is blue-yellow deficiency. Okay? And if the person is having optic nerve disease, red-green deficiency is more common. Again, please mark it as a question. If it is retinal diseases, blue-yellow. If it is optic nerve diseases, red-green. And if it is congenital, it is X-linked recessive, seen in males. Okay, so these are the simple facts to be remembered here. Now, how to test the color vision? Now, this is again a good high-yield topic here. The color vision testing is done by different methods. The common methods or the common tests which are done for the color vision testing, they are, friends. Please have a look here, the names. First is the test which is called as pseudo-isochromatic plates, and the commonly used is Ishihara plates. Ishihara plates. Okay. The second test is known as Farnsworth-Munsell 100 hue test or FM 100 hue test. FM, your FM radio for color vision. Farnsworth-Munsell 100 hue test. Then there are enamelloscopes to check the vision, color vision. And then we have got the test, lantern test or wool test, which are again done for the color vision. Lantern test is specially done for the railway drivers, the train drivers. Lantern test. So you can imagine the red and green lantern, for them it is important to know. Okay, so these are the important color vision testing. Again, names to be remembered: Ishihara plates, FM 100 hue test, enamelloscope, lantern test, which is done for railway train drivers, and wool test. Okay. So how do the Ishihara plates they look like? So Ishihara plates, the question can come as image question. They can show you these and to identify. So these colored plates, isochromatic plates in which number are written. So if you are able to identify the number, so you must have seen even sometimes the WhatsApp messages, they come, check whether your vision is normal or not. So you can see these numbers are written 12, 42, 74, 6. So you can check your vision also whether you are able to identify these numbers or not. So image-based question based upon these Ishihara plates. Image-based question based upon Ishihara plates. Okay, right? Then this is just for your understanding how this 100 hue looks like. Even if you don't remember the image, it is fine. So you can see 100 shades. These 100 shades are there. Farnsworth-Munsell 100 hue test. So this is how this test looks like. Okay. Now, another thing to be remembered about these tests is, so come to the next page there. The most commonly used test for screening, okay, most commonly used test for screening is Ishihara. So this can be question. Most commonly used screening test is Ishihara, which we do in the OPTs also. Okay. Then most specific test. So any idea which can be the most specific test for color vision? That is your FM 100 hue test. This is most specific. Okay. This can be asked as a one-liner question. And if you want to quantify, like, how much quantification, then again, we can use two tests. One is FM 100 hue test and enameloscope. Ishihara is qualitative. But if you want to do quantification, FM 100 hue test and enameloscope. Okay. So these are the points for color vision. So color vision, a simple thing to remember, but a one-liner question can come. So name of the test we have done. Remember the image of Ishihara plates, beta. Don't forget the image of Ishihara plates. Simple to remember. Once you see, now you know these are Ishihara plates. And few important points. The most common screening test is Ishihara. Most specific test is FM 100 hue. And if you want to quantify, for quantification, FM 100 hue and enameloscope. Okay, right? Right. Then in the physiology, the last thing which we need to know to make, like, for our further chapters, the concept of visual field. The concept of visual field. So I want you to focus here because this visual field will be used a lot for your glaucoma, for your neurology, neuro-ophthalmology. So have an understanding here. So first of all, write down the definition of visual field. Write down the definition of visual field. So the definition is, please write it down. Visual space which we see around us when the eye is focused on central fixation point. This is the definition. Visual space which we see around us when the eye is focused on central fixation point. Okay. So you have written this. Now, first of all, understand everything is written also there. You know, both your eyes, you are looking straight ahead, but still you can see what is on the sides. You can see what is below, what is above, even when you are looking straight. Okay. So not only we are able to see the object which we are focusing, we are also able to see the visual space around us. We are also able to see the visual space around us. So that whole visual area is known as visual field. Field means space. Okay. Now in the visual field, there will be a central point. Okay. In the visual field, there will be a central point where you are looking. That central point is called fixation point. And then the whole area around it, around the fixation point, we will call it as visual field. Okay. So as you can see, you can divide the visual field. There will be upper visual field, lower visual field. Okay. Temporal visual field, that is on the outer side. Nasal visual field, that is on the inner side. Okay. So in the center is the fixation point. In the center is the fixation point, and then 4 quadrants, you can say: upper, outer, temporal, that is on the outer side, and inner or on the nasal side. Okay. And you can divide it also into meridian. That, you know, there is this vertical meridian passing through the fixation point and a horizontal meridian passing through the fixation point. So this is how we present the visual field graphically. Okay. Central area, the whole space around that, and 4 quadrants, you can say. Okay. Now the central area, the fixation point, corresponds to what we call as fovea. Okay. So look here, the fixation point in the visual field corresponds to fovea. What is fovea? We'll be discussing in retina also. If you want to write down here, you can write down. Fovea is the central most part of retina. Fovea is the central most part of retina. So the fixation point corresponds to fovea of the retina. Okay. Then in the visual field, there is what is known as blind spot. Then in the visual field, there is a point known as blind spot. What is blind spot? Blind spot is an area in the visual field where you cannot see the image. Where you cannot see the image. And where you cannot see the image, that area is known as absolute scotoma. That area is known as absolute scotoma. And this area corresponds to area of optic nerve. Okay. So in the visual field, there is a central point. That central point is called fixation point. Fixation point corresponds to fovea. And then there is another area in the visual field which is called blind spot. The blind spot corresponds to optic nerve. Okay, so this can be a one-liner question. So you can mark it like this. And what is the position of the blind spot? Fixation is in the center. The blind spot is temporal. So fixation is in the center. The blind spot is always temporal. Okay. So you see here, this is the blind spot, temporal, on the right eye. And this is the blind spot on the left side. Okay. So simple thing to remember, beta, but this can be asked as a question. The blind spot is on the temporal side of visual field and it corresponds to optic nerve. So something regarding scotoma. Scotoma word is used for the field defect. Okay. So you can write it down here. Scotoma means a defect in the visual field. Defect in visual field. Okay. Scotoma means defect in visual field. Now, scotoma can be positive scotoma when there is something abnormal in the visual field. For example, floaters. Floaters are the shadows which sometimes we see. Okay. Which are actually not there in the outer atmosphere, but we are seeing those shadows. They are known as floaters. Those shadows are known as positive scotoma, that you can see them. They are physically not there, but you can see them. They are known as positive scotoma. And then there can be negative scotoma, that there is physically something present there, but your eye is not able to see that. So if there is absence of visibility of something, that will be known as negative scotoma. Okay. So visual field defects, they can be positive scotoma, for example, floaters, or more commonly negative scotoma, that is there is something there, but eye is not able to see that particular area. That is absence of visibility. That will be negative scotoma. Okay. For example, blind spot. Blind spot is a negative scotoma. Blind spot is a negative scotoma. Floater is a positive scotoma. Okay. And then in the last, what are the normal boundaries of visual field? Okay. It is not that visual field is unlimited. There are normal boundaries of the visual field. Now, in the visual field, the largest area or the biggest area is your temporal, which is about 100 degrees. So on the outer side, you can see the maximum, okay, 100 degrees. Then inferior field is 75 degrees. Okay. Nasal field is 60 degrees. The superior can be, please add over here, 50 to 60 degrees. Please write here, because some books, they mention superior and nasal same as 60 degree. Some books, they mention superior as 50 to 60 degree. So I hope you have added here superior 50 to 60 degree. So which is the smallest field? Which is the smallest field in a normal eye? Superior. Okay. So sometimes the question can come, the smallest visual field will be, you can write it down also, superior, and largest will be temporal. Okay. So these are the normal boundaries of the visual field. Smallest visual field will be superior and the largest visual field will be temporal. Okay, right? You're another one-liner for you. Okay, right. Then what are the tests for the visual field? So everything which is there in the eye, we can measure that. We can test the function of the eye. which is very remarkable thing, isn't it? So the visual field testing is known as perimetry. Okay, so please remember this term, perimetry. Again, a one-liner. What is perimetry? The testing of the visual field is known as perimetry. Now, there are three types of perimetry. One is known as kinetic perimetry. Kinetic means movement. That is, you show the target to the patient, which is moving from out to in, from above to below. Okay, so here the target is moving. Where the patient is able to see the target, that will be the boundaries of his vision field. Okay, so kinetic perimetry means target is moving. There are two examples of kinetic perimetry. One is confrontation method. Other type is Goldman perimeter. So please remember the names. Confrontation method, in which we show the finger to the patient. Okay, not like, abuse the patient. We show finger, whether the patient can see the finger or not. Okay. Then the second one is your Goldman perimeter, which also uses a moving target. So these two are examples of kinetic perimeter. Okay. The second type is static. Static means stationary. Here the target is stationary. We change the brightness of the target and we judge whether the patient is able to see that or not. Okay. In static perimetry, the brightness of the target is changed. It is stationary target. Examples are Humphreys perimeter, octopus. So again, please remember the names. So all this about these points about perimetry, they can be asked in question. Humphreys perimeter, octopus, they are examples of static perimetry. Okay. Then a third type is known as microperimetry, in which we test only the small area, the macular area of retina. Only the small area is tested. Okay. That is known as micro. Micro means small. Small area is tested. Just the macular area of the retina. Okay. So three types of perimetry. Then an image-based question can be asked. What is this image? What is this image? Now, this is the example of the Humphreys visual field. Humphreys visual fields. Okay, I told you, Humphreys perimetry, static perimetry. So when we do the test on the Humphrey visual field, which is most commonly done, this kind of chart will represent the visual field. So this can be asked as question, or when you get your neuro-optha question, they can show you visual field like this. So please remember the image. This is Humphreys visual field. Okay. So if I show you this part of the field, so here you can see this is your central point, central fixation. Okay. This is your central fixation. This is central fixation. What is this? Any guesses, friends? What is this? This is your blind spot. This is your blind spot. Okay. And what are these? These are your scotomas. That is the field defect. Okay. So this is how we see clinically when the patient does the visual field test. Okay. There will be central fixation point. There will always be a blind spot due to optic nerve. And then if there is any abnormality, you can see scotomas like this. Right? So got the thing? Right. So the chart you can identify now. This is Humphrey's visual field chart. Humphrey's visual field chart. Fine. So with this, we have covered most of the important topics which we need to know about the vision testing. Okay. So I hope you have marked the important points, highlighted the important points. When you revise, please try to remember those points. They are your high yield points, friends. Now I'm going to go to the next thing in which I'm going to, that is more for your concept building, for your understanding. Okay, that when we examine the eye, how we examine the eye, or how the eye looks like when you examine it clinically, because that will help you to understand various clinical signs in ophthalmology. Right? Various clinical signs in ophthalmology. So, okay, so before that, I move on to that. Would you like to do some quick workbook questions based upon the anatomy and the physiology? They are just basically to give you an idea of revision. Okay? So if you want to do the quick workbook questions, you can see here. The first question is primary visual area in the cortex. So we did in our visual pathway, the primary visual area in the cortex is area number 70. Okay, right? Then second question is avascular coat of the eye. So I told you which out of these, which part of the eye is avascular. Uvea, retina, cornea, choroid. So if you remember, it is your cornea, which is the avascular coat. Okay. This is just to revise the points. Then aqueous humor is secreted by. Remember the fluid which was there in the anterior and posterior chamber. Aqueous humor is secreted by, yes, pars pleccata. Pars pleccata. Okay, right? This I have not told you as yet, the refractive power of the eye, but we'll do it later. But you can mention refractive power of the eye is 60 diopters. 60. Then we did the visual pathway, though again we will be doing it, but here the parts which we saw in the figure of the visual pathway, which of the following is not the part of visual pathway? Optic nerve, optic tract, medial geniculate body, optic radiations. So the right answer here is medial geniculate body is not the part. It is the lateral geniculate body which is the part. Okay? So these kind of one-liners, it is just for your revision so that you understand them. Okay, right? Okay, so then we are going to the next chapter, which I have labeled as clinical ophthalmology. So the first picture is telling you about what I have already told you, the different parts of the eyeball. I'm not going to kind of do it again. It is a labeled picture. You can see that. What I'm going to concentrate now is that when you examine the eye, how do you look for the normal structures on examination so that we can pick up the signs in the diseases. Okay, right? So when you think that a patient has come to you in the OPD and you are going to examine the eye. Now, when you want to examine the eye examination is divided into three parts. External examination of the eye, what is there on the surface, examination of the interior segment of the eye, and examination of the posterior segment of the eye. Okay, so that completes our eye examination. So what we do in the examination of the external eye. Now in the external eye, you can do the examination even with the torchlight. Okay, so the external surface of the eye can be examined even with the torchlight. You shine the light on the eye, you see the eye has got the lids, the upper lid, the lower lid, where these two areas, they join each other. That is known as medial canthal angle and lateral canthal angle. Okay. And on the eye surface, what you are seeing? On the eye surface, you can see this transparent cornea. The cornea will be transparent. Okay, right? Then you see the white area, which is your sclera, right? The junction of the two, limbus. Junction of cornea and sclera, limbus. Okay. Then because the cornea is transparent, through the transparent cornea, you can see this brown iris. Because cornea is transparent, through the cornea, you can see brown iris, brown or blue or gray or whatever is the color of the iris. Okay. And in the center of the iris, you see this aperture or opening, which is pupil. So this is how the normal pupil will look like. Okay. Then sclera is covered by another membrane, which is called conjunctiva. We are going to learn next about conjunctiva only. So on the surface of the sclera, you will see a membrane, which is called conjunctiva. Okay. And here, just even if you don't remember here, it's fine. These inner area, this inner raised area is known as caruncle, and this fold is known as plica semilunaris. This is just for knowing it, even if here it doesn't have the importance there. So this is the surface of the eye. You can see even on torchlight. So you can see whether there is any abnormality of the lids. You can see whether the cornea is clear or not. You can see how the sclera is. You can see how the eyelashes are. You can see the color of the iris. You can see the pupil. So this is done on external examination. Okay, right? Now I want to see the more detailed structure, more detail. So if I want to do the more detailed structure, I will do the examination of the eye by an instrument which is known as, you can write it there, slit lamp biomicroscope. Slit lamp biomicroscope. Okay, so there is an instrument, slit lamp biomicroscope, through which we can examine the anterior segment of the eye. Now, when we do the slit lamp biomicroscope checkup, it gives me optical sections, slit. It gives me optical sections of different structures. Okay, so this is how the eye will look. And when you get an image-based question on the eye, how will you identify these structures? On slit lamp biomicroscopy, friends, please concentrate on this image here. It is important for your future chapters also. So when I am looking through the slit lamp, so first of all, again, I can have a look, more detailed look at the lids. I can have a look at the sclera. Okay. Then I go now section wise, from front to back. I go now section wise. So what are you seeing here? This is the section of cornea. Okay. This white section is your transparent cornea. So first section will be of cornea, right? Then you will be seeing the iris through the cornea. Again, just like torchlight, you will be seeing the iris. Okay. Then this section, this is your lens of the eye. This is how the lens will look like. Okay. Behind the iris will be lens. So this will be the anterior part of the lens. This is the posterior part of the lens. Okay. And what is between Iris and cornea, iris and cornea, anterior chamber. Okay, so you can see the anterior chamber also between iris and cornea, right? So this space is filled with the fluid, the aqueous, remember? So you can see that. And then you can see the part behind the lens also. So part of the 1/3 vitreous cavity also you can see. 1/3 vitreous cavity. So this is what you are seeing when you are looking at the eye through slit lamp. And here we can see if all the structures are normal or abnormal. Okay, so the first slit will be of the cornea. Then through the cornea, you can see the iris. Then the next slit will be, the bright slit will be of the lens. Then between cornea and iris will be again the anterior chamber filled with the aqueous fluid. And behind the lens will be vitreous. You cannot see whole of the vitreous cavity, 1/3 of the vitreous cavity. Okay, so now you know how to look for different structures if there is an image like this. Got it, beta? Right? Right. Now, this is the anterior segment examination with slit lamp. If I need to now, so already I have mentioned you, this is all written also, so you don't need to write anything. What all structures you can see through the slit lamp examination. Now, when I want to do the posterior segment examination, that is behind the lens now, the posterior segment of examination will make me look at the retina, optic nerve, and under the retina, I can see the choroid also. Okay, so this is how when we examine the posterior segment, it is also known as fundus examination. Okay, another term for you, the examination of the posterior segment is also like, it is fundus examination. And when we are doing the examination of posterior segment, this is how it looks like. This is how it looks like. So what all structures we are seeing here, we will be again doing it in the retina chapter, but here giving you a brief introduction. But all you are seeing here is, this is your optic nerve. This is your optic nerve. Okay. This central area is your macula, the central part of the retina, macula. And here in the center of this macula is your fovea. So these are the terms we will be using in different diseases. You can see the optic nerve, the central part of retina, macula. In the center of macula, the area which is called fovea. And you can also see these are retinal vessels. These are retinal vessels. Okay, so this is how I am doing the fundus examination. We will be discussing it later also in retina chapter. So what is the instrument which is used to do the posterior segment examination? So everything is written in your workbook. So this again can be as one-liner question also. We do the instrument which we used are known as ophthalmoscopes, ophthalmoscopy. And the fundus examination is also called as fundoscopy. There are two types of the ophthalmoscopes. We will be discussing instruments afterwards. Here the introduction. One type of ophthalmoscope is known as direct ophthalmoscope. Direct ophthalmoscope gives me only central retina can be seen. Okay. And the second type is your indirect ophthalmoscope. And in indirect ophthalmoscope, I can see all these structures. I can see optic nerve, I can see veins, arterioles, central retina, mid-peripheral retina, and peripheral retina till its boundary. The boundary, as you know, ora serrata. So I can see till that. Optic nerve, optic veins, central retina, as well as peripheral retina. So these are the two instruments which we use: direct ophthalmoscopy, indirect ophthalmoscopy. Okay. So we'll be discussing the instruments in the last also. So this is the brief now examination procedure. How to check the eye: diffuse torchlight examination, slit lamp examination, and ophthalmoscopy. So now all of you are ready to examine your patients. Okay, right? Okay. So let's take a pause of a minute or so, because now we are going to move on to the diseases of different eye structures which lead to different conditions. Okay. And next, the first one which we are going to discuss is about conjunctiva and diseases of conjunctiva. Now, in most of the exams, I have seen about 2 or 3 questions coming from the chapter of conjunctiva. So all of those who want to prepare exam oriented, the chapter of conjunctiva is simple. Okay, it is a simple chapter, easy to understand, high yield, because there is always nearly 2 to 3 questions from this chapter. Okay, so let's start with conjunctiva. Take a few deep breaths. I can take a sip of water, and we start with conjunctiva. Just let me put my iPad to charging also. Right. So coming to the chapter of conjunctiva, diseases of conjunctiva. The anatomy which we need to know. So what is conjunctiva, friends? I mean, how do we kind of identify conjunctiva? So conjunctiva is a translucent layer. Okay, it's a translucent layer which is covering the part of the sclera, okay, as well as the under surface of the eyelids. Okay. So please write it down. What is conjunctiva? Please write it down. So conjunctiva is a translucent layer which covers anterior sclera, that is the front part of sclera, anterior sclera, and lines the eyelids. The upper surface of the eyelids is covered by skin, okay? And the under surface of the eyelid is covered by conjunctiva. So it is a translucent layer. Now, conjunctiva can be divided into three parts. Conjunctiva can be divided into three parts. So this figure shows you three parts of the conjunctiva. These three parts, they are known as the bulbar part, fornical part, and palpebral part. Okay. Bulbar, fornical, and palpebral. Bulbar is which is covering the sclera, this part. This part, which is covering the sclera, is your bulbar part. Okay. Then palpebral, which is covering the lids, under surface of the lids, is palpebral. Palpebral word is used for lids. So which is covering the lids is palpebral. Then if you notice, like, you must have noticed in your eye, there is a gap between the eyeball and the lids. It is not adherent. There is a gap between the eyeball and the lids. So that gap is called fornix. The gap between the eyelid and the eyeball is called fornix. And the conjunctiva, which covers the fornical area, is the fornical conjunctiva. Okay, so it's kind of helping you to join the lids with the eyeball. Okay, so you got the idea of the anatomy. The part of the sclera which is covered by conjunctiva, that conjunctiva is bulbar conjunctiva. Then it is lining the lids from inner side, that is palpebral conjunctiva. And the fornical area between the eyeball and the lid, that area is also covered by conjunctiva. The conjunctiva is reflected in those fornices. That is called fornical part of the conjunctiva. Okay. Now, in the anatomy of conjunctiva, please remember the two important points. The conjunctiva has got two parts in the anatomy or histology. So if I talk about histology, you can label it as histology. You can write here in the space provided here. So histology, the conjunctival epithelium, the outermost will be conjunctival epithelium. Now, this is important point to remember. The conjunctival epithelium is non-keratinized, stratified squamous epithelium. Okay, so this can be asked as a question. What is the nature of the conjunctival epithelium? Non-keratinized, because if it is keratinized, it becomes skin-like. It is non-keratinized, stratified squamous epithelium. Okay. Then the second part is your stroma. The conjunctival stroma. Please write it down. The stroma has adenoid layer which contains the lymphoid tissue. Okay. It has adenoid layer which contains lymphoid tissue. Right? So these are the 2 points to be remembered about conjunctiva. What is the nature of the conjunctival epithelium? Non-keratinized, stratified squamous. And the stromal layer has adenoid or lymphoid tissue, because conjunctiva is on the surface. So that has got a protective role. If there is anything, any injury, it protects because it has adenoid layer or the lymphoid tissue. And the third thing is there are few specialized cells in the conjunctiva. Few specialized cells in the conjunctiva, what is known as goblet cells. What is the function of this goblet cells? Write down, they secrete mucin, M-U-C-I-N, mucin layer of tear film. You know, your eyes are always kept moist by a tear film. The eye surface is always kept moist by a tear film. Tear film has different components. We will come to that in the coming chapters. One component is mucin layer. Now, this mucin layer is secreted by goblet cells, which are present in bulbar conjunctiva. Now, this can be a one-liner question. This can be a one-liner question. Where are goblet cells? They are present in bulbar conjunctiva. Which part they are maximum? In this nasal part towards the nose side. They are maximum in number in nasal part. What is the function of the goblet cells? They secrete the mucin layer of tear film. They secrete the mucin layer of the tear film, which helps in keeping the eye moist. Okay, friends, right? So goblet cell question is important one-liner for you. Now we come to the diseases of the conjunctiva. Now, in the diseases of conjunctiva, the most common is what is known as conjunctivitis. Okay. Conjunctivitis means inflammation of the conjunctiva. Itis anywhere in the body means inflammation, arthritis, or some hepatitis, or whatever. Itis means inflammation. Now, in the inflammatory diseases of conjunctiva, which will be conjunctivitis, there can be two etiologies. What are these two etiologies? One, it can be allergic. The second etiology can be infectious. Okay, so two etiologies of conjunctivitis: allergic or infectious. Now, in the conjunctivitis, there can be common symptoms and signs. What are the common symptoms and signs of conjunctivitis? Okay, so let's understand this. Now, in the symptoms, the most common symptoms, there are 5 more common symptoms. Whenever there is conjunctivitis, there will be inflammation or dilation of the vessels. So the eye will appear as a red eye or pink eye. Okay? So conjunctivitis is a cause of red eye or pink eye. Pink means when it is less red, mildly red. That will be pink eye, friends. Okay? Now, whenever there is conjunctivitis, there will be discomfort, that you will feel that, eye is not comfortable, discomfort or mild pain. But remember, it will never be very painful. Okay? Never, you can write also here, never very painful. It is more of discomfort, because that will help me in differentiating conjunctivitis from other diseases in which there can be red eye. So there can be red eye, but mild pain or just the discomfort, friends. Okay. Then there will be a sensation of grittiness. Grittiness means irritation, as if something is in the eye. Grittiness. There can be photophobia, that a person dislikes the light. Bright light will not be comfortable, hurting. And very importantly, there will be watering and discharge. The eye will be watering or sticky. Okay? So why these symptoms are important? Because they will help you solving clinical case scenario questions. Okay? So please focus here, friends, the symptoms of conjunctivitis, because they will help you solving the clinical case scenarios. What can be the symptoms? Red eye or pink eye, mild pain or discomfort, photophobia, grittiness or irritation, watering and discharge. Okay, right? Then moving to the signs. What signs you will see? So coming forward, clinical signs. So first of all, there will be conjunctival congestion. That is, the eye is appearing red. Now this conjunctival congestion will be superficial in nature because conjunctiva is the superficial layer. And it will be more common in the fornices. You know, this area is appearing whitish, not much congestion, but more in the fornices. So this will be the sign of the conjunctival congestion. It is superficial, more in the fornices. Okay. Then discharge is very, very important because with the help of discharge, you can identify the etiology of the conjunctivitis. From the discharge, you can identify the etiology. If it is a watery discharge, etiology is viral, friends. Okay. In viral conjunctivitis, you will have watery discharge. If it is mucoid discharge, then the etiology is allergic. This type of discharge is also called ropey or stringy discharge, like a string. Ropey, that you can pull the discharge like a rope or a string. This is highlight of, please mark this, the allergic conjunctivitis. Okay, important point. Ropey, mucoid or stringy discharge, allergic conjunctivitis. Okay. Then the mucopurulent type of discharge, etiology can be bacteria or chlamydia. Okay. And if it is markedly purulent, that indicates severe bacterial infection. So discharge is very, very important in identifying the type of conjunctivitis. That will help you solving the clinical case scenario. Please remember, once again, I'm telling watery in viral, mucoid, which can be also called as ropey or stringy in allergic, mucopurulent in bacterial or chlamydia, purulent in severe bacteria. Okay, friends? Then another sign which is seen in the conjunctivitis is what is called as papillae. So what are papillae? So you see this figure here and you see these lesions on the undersurface of the lids in the palpebral conjunctiva. These lesions are your papillae. Now, if you notice carefully, these papillae, they have got a red center. And they are flat topped. Okay. So this is how you identify papillae. Okay? So you can write it down also here. The papillae, they have fibrovascular core. So they are red in center. And they are flat topped. Pitchke hue. Okay. Now, papillae, if they are very big in size, like this one, they are then known as giant papillae. When they are big in size, they are known as giant papillae. And then they give an appearance to the conjunctiva like a cobblestone. You must have seen the cobblestone pathways. Okay. So it looks like a cobblestone. So this again can be a question. The cobblestone appearance of the conjunctiva is due to giant papillae. Okay, please highlight it. The cobblestone appearance of the conjunctiva is due to giant papillae. When you see the papillae, it indicates two kinds of etiologies. Okay, the etiology is mainly, most commonly, they are seen allergic conjunctivitis. Okay, so they are seen in allergic conjunctivitis. They can also be seen in bacteria, and another important is contact lens use. Okay, so please highlight these 2 points: allergic conjunctivitis and contact lens use. You will see papillae. Again, in clinical case scenario, this thing can come if there are seen papillae, if there is ropey discharge, then etiology is allergic. If a person is using contact lens, then again there can be giant papillae there. So that indicates the patient is having allergy due to contact lens use. That also gives a type of conjunctivitis. Okay. So please remember once again this picture of the papillae also. This image I will show you in allergic conjunctivitis also. How the papillae they look like. Giant papillae, they give cobblestone appearance to conjunctiva. And 2 important conditions where the papillae will be seen is allergic conjunctivitis and if a person is showing reaction to contact lens use. Okay, friends, you will remember this. Okay. Okay, coming forward there, another sign which you can see is the formation of what is known as follicles. Okay. Now follicles, have a look here. These round, look at the figure here to identify the follicles, these round lesions. Now you see these lesions, they have white center. They have white center. So differentiation from papillae. Papillae was red in center, flat topped. These are white in center and they are round like follicles. Okay. So follicles, you can write it down here also. The follicles, they are white in center. Please write down white in center and they will appear round, not flat top. What are they? They are the aggregation of lymphocytes. The follicles are aggregation of lymphocytes. The 2 etiologies in which you will see follicles, one is the viral conjunctivitis and then chlamydia, for example, trachoma. So if in the history or if in the case scenario it is mentioned that follicles are seen, so think of viral or chlamydial infection. And how do you differentiate follicle from papillae? I have told you. Okay. Follicles are white in center and they are roundish. They are not flat topped. Okay, friends, right? Then in conjunctivitis, you can also see that there is the formation of a membrane on the surface. Now, this membrane can be a true membranous conjunctivitis, which is seen in the cases of Corynebacterium diphtheria. If the conjunctivitis is due to diphtheria, there will be a true membrane formation, as you are seeing here, a membrane formation. Okay, or it can be a pseudo membrane. It can be a pseudo membrane, which can be seen in any severe bacterial conjunctivitis. The pseudo membrane is just the organized exudates. Pseudo membrane is nothing but organized exudates. And how we differentiate a true membrane from pseudo membrane? The pseudo membrane can be peeled off easily. The pseudo membrane can be peeled off easily, whereas a true membrane will bleed if you peel it off. It bleeds on peeling. If you try to peel it off, it will bleed because it is the part of that tissue only. Okay, and a cause of the membranous conjunctivitis is, again, important point, Corynebacterium diphtheria. Okay, right? So these are the important signs which we have seen, which you will find in a case of conjunctivitis. So please see once more, there will be conjunctival congestion. Okay. There will be discharge, different type of discharge. There will be papillae. There can be follicles. There can be membrane formation. Right? So if in the case scenario, any of these is there, you can think that it can be conjunctivitis. Now we are going to learn the features of specific type of the conjunctivitis. And first of all, let us discuss infectious conjunctivitis. Infectious conjunctivitis. Now in infectious conjunctivitis, first of all, the infection can be due to virus. The infection can be due to viral, and in viral conjunctivitis, what are the features of the viral conjunctivitis? The viral conjunctivitis is mostly mild type. Okay, it is mostly mild, and it is leads to what we call as pink eye. It is not that severe. So the redness will be like, pinkish. Discharge, watery. Okay. Pink eye, watery discharge. Sometimes this condition is also called as Madras eye, though it is not commonly now called it like this. Right? Madras eye, because there was epidemic in Madras conjunctivitis, that the way it's called Madras eye, if you think of that term. But mostly we call it as pink eye. And it is mostly self-limiting. You really need not give any treatment to the patient. Most common cause of viral conjunctivitis is adenovirus. So please remember this. There can be other viruses. The names are written: enterovirus, echovirus, Coxsackie virus. But the most common, please remember, the most common cause is adenoviral for the viral conjunctivitis. Okay, right? Now, in the adenovirus can cause two kinds of conjunctivitis. One is known as epidermic keratoconjunctivitis, EKC. So as the name is suggesting, okay, so try to remember the things from the name of the condition. Now, epidermic conjunctivitis means it will come as like, epidemics. You must have seen eye flu chala hua hai, eye flu phaila hua hai. When we call it as eye flu, it means it is epidemic. It is very easily spreads from one person to another person. So epidemic keratoconjunctivitis is caused by adenovirus. Such patients, they can also have lymphadenopathy. There can be subconjunctival hemorrhages. Already I told you the follicles are formed. In viral conjunctivitis, follicles are formed. Okay? And in 80% of the cases, it causes nummular lesion. Nummular means, please write it down here, they are subepithelial corneal lesions. So though it involves the conjunctiva, but in the cornea, subepithelial lesions, they occur. So that is why kerato means cornea. So when it is epidemic keratoconjunctivitis type, the cornea is also involved, but subepithelial lesions. Okay. So these are the features of epidemic keratoconjunctivitis. The point to be remembered, question can be asked, epidemic keratoconjunctivitis is caused by, you can cause will be adenovirus. Okay, friends, the cause will be adenovirus. Then the second type is called pharyngoconjunctival fever. This is again caused by adenovirus. Now, pharyngo means pharynx is also involved. So, The features are pharyngitis. If there is pharyngitis, there is fever, conjunctivitis. In these cases, corneal invasion is only in 30%. So from the name, you can remember. You don't even have to memorize. If it is epidemic keratoconjunctivitis, cornea is involved in 80% cases, subepithelial lesion. If it is pharyngoconjunctival fever, cornea is 30% involved and it has pharyngitis. Both types, they can be caused by adenovirus. So that is the question. Pharyngoconjunctival fever is caused by adenovirus. Epidemic keratoconjunctivitis is caused by adenovirus. Okay. Now, sometimes the viral conjunctivitis can also be very severe. So I told you mostly viral conjunctivitis is mild, pink eye, self-limiting. Okay? But sometimes it can be severe and it can lead on to conjunctival hemorrhages. When it is severe and it leads to conjunctival hemorrhages, then it is called acute hemorrhagic conjunctivitis. Then it is called acute hemorrhagic conjunctivitis. Okay, so the image is shown you here. This is just for your reference. I will tell you, friends, which image can come as image-based question. Some of the images are for your concept building. Okay? Because if you know how it looks like, you can always remember. The visual memory is always better than the reading the words or listening the words. That is my idea. Okay? So I have put a lot of images. Some are for your understanding. Which image can come as image-based question? I will keep telling you. I have been telling you side by side, isn't it? Right? So when it is a severe viral conjunctivitis, it causes conjunctival hemorrhages. That is called acute hemorrhagic conjunctivitis. The question to remember over here is the causes. The causes of acute hemorrhagic conjunctivitis. The most common cause is enterovirus 70. So this can be asked as a question. Please mark it. The most important cause or most common cause of acute hemorrhagic conjunctivitis is enterovirus. Okay? There can be other causes. There can be other causes. We can write it down. Coxsackie. Coxsackie. Oh, I'm forgetting the X over here. Coxsackie virus. Echovirus. Coxsackie virus, echovirus, and even adenovirus. So even if you don't remember the last three, please remember the first one. The most common cause of hemorrhagic conjunctivitis is enterovirus 70. Okay. Now in the viral conjunctivitis, come back to this space over here. Few points to be written about COVID-19 conjunctivitis. So if you know the COVID-19 infection was discovered by an ophthalmologist in China. Now it is a history, you can say. So how the COVID-19 came into being, because an ophthalmologist found conjunctivitis and then it was related to COVID-19. Okay. So few points regarding COVID-19 conjunctivitis. Conjunctivitis is the most common eye involvement in COVID-19. Okay, so please write down most common involvement is conjunctivitis in COVID-19. Most common involvement is conjunctivitis. Okay. And which type of conjunctivitis? Viral, we know. Which type? Follicular. Follicles are formed. Follicular conjunctivitis. So both of these, they can come as question. Which type of conjunctivitis is seen in COVID-19? Okay. So question, which type of conjunctivitis is seen in COVID-19? It is follicular type of conjunctivitis because COVID-19 is a virus. Okay. What is the most common involvement in the eye in a case of COVID-19? It is conjunctivitis. Okay. Not any retina disease, not any optic nerve disease. They can be there, but the most common is conjunctivitis. Okay, so these are two questions related to COVID-19 and conjunctiva and eye, you can say. Okay, right? Okay, then coming on to the next type of the etiology is your bacterial conjunctivitis. Now we know the bacteria, they are associated with purulence. Okay, so bacterial conjunctivitis is more severe than the viral one because it is associated with purulent kind of features. In the causative organisms, the most common, so this can be asked as a question, the most common cause of bacterial conjunctivitis. So please, wherever I have mentioned most common, highlight it, and that is the point to be remembered. You can leave the rest of the causes. You can complete the list, but please remember the most common causative organism. So most common causative organism in bacterial conjunctivitis is Staph aureus. Okay. So what are the other causes? You can complete the list. Pneumococcus, H. influenzae, and Moraxella catarrhalis. This is to complete your list. Common bacteria. Staph aureus is most common, that is to be remembered. Rest are pneumococcus, H. influenzae, Moraxella catarrhalis. Okay. Now in bacterial, we know there will be pus. So you can see the viral, it was just a pink eye. So in bacterial, there will be more redness, red eye. There will be mucopurulent or purulent discharge. There will be more discomfort. Okay. In bacteria, there can be certain bacteria which are very, very severe. They cause very severe conjunctivitis. Okay. So they can cause membranous conjunctivitis. Already I told you, membranous conjunctivitis, the common cause, diphtheria, even gonococcus can cause membranous conjunctivitis. Okay. So membranous conjunctivitis will be caused by diphtheria. So when it is severe, then you have to give systemic treatment also. Okay. Normally, viral, self-limiting, no treatment is needed. For bacteria, you will give topical antibiotics. Topical antibiotics. Topical means eye drops. Okay. So for bacteria, you will give topical antibiotics, that is the antibiotic eye drop. Okay. If it is very severe conjunctivitis, then you have to give systemic treatment also. Like for diphtheria, you need to give anti-diphtherial serum. Similarly, for gonococcus, you have to give, I will just now coming to the gonococcal conjunctivitis, because gonococcal conjunctivitis can be very severe. So few points about gonococcal conjunctivitis. It can be very severe conjunctivitis, very severe. what we call as hyperacute conjunctivitis. So there can be question, hyperacute purulent conjunctivitis is caused by gonococcus. So please remember, gonococcus is not a good news for eye. Hyperacute conjunctivitis. It can cause lymphadenopathy. And most seriously, it can cause corneal ulceration and corneal perforation. So this is the important point. That is why gonococcal conjunctivitis is dangerous, because it can cause corneal ulceration and corneal perforation. The gonococcus can penetrate intact cornea, even when the cornea is not injured. The gonococcus can penetrate intact cornea. Okay, so please remember, the gonococcal conjunctivitis has to be dealt seriously because it has various complications. It causes hyperacute conjunctivitis. It can cause corneal ulceration. It can cause corneal perforation. Right? So that is why whenever there is gonococcal conjunctivitis suspected, then the treatment will include topical antibiotics, cycloplegics like atropine, as well as systemic antibiotics. So we have to give all three: topical antibiotics, atropine, and systemic antibiotics. For which type? Gonococcal type, because gonococcal is very, very severe. Okay. So in 15 minutes, my friends, we will be breaking for the lunch. And because I just want to go through conjunctivitis a bit here. Okay, is that all right? In 15 minutes, we will be breaking for lunch. Okay. So adult chlamydial conjunctivitis, the next. So in bacteria, you have discussed the normal bacteria and the gonococcal. Now coming on to the chlamydial conjunctivitis. Now in chlamydial conjunctivitis, this type which we are discussing is adult chlamydial conjunctivitis. It is also called swimming pool conjunctivitis. Now these can be your one-liner questions, that the causes of swimming pool conjunctivitis. So it can be caused by Chlamydia trachomatis, which serotypes? D and K. It is important to remember. Chlamydia D and K serotypes, they can cause swimming pool conjunctivitis. And the second cause of swimming pool conjunctivitis is adenovirus. Adenovirus. Okay, right? So why are we calling it swimming pool conjunctivitis? Because again, it spreads from person to person. Public spread is through the, if you use public swimming pools, it tends to spread. So that is why the name swimming pool conjunctivitis. So one-liner question, causes of swimming pool conjunctivitis: Chlamydia trachomatis serotypes D to K and adenovirus. Okay, friends? Then another specific type of conjunctivitis which can be asked in your exam is angular conjunctivitis. Angular conjunctivitis. Another name of the angular conjunctivitis is diplobacillary. Why it is called so? Because the causative organism is Moraxella Axenfeld, which is a Diplobacillus. Diplobacillus. Now this can be asked in the question. Okay. Now all these 4 or 5 points which I have written, all of them, they can be asked as a question. Question: what is the cause of angular conjunctivitis? So the organism, now you know, Moraxella Axenfeld. Okay. Then it can be asked, what is the cause of diplobacillary conjunctivitis? Again, the same thing, Moraxella Axenfeld, because it is a diplobacillus. Why it is called angular conjunctivitis, or what is the special feature of this type of conjunctivitis? You see, it causes excoriation. Eye is red more at the angles, outer and the inner canthi, and there is an excoriation of the skin. The skin also becomes excoriated. Why it happens? Because this bacteria produces a proteolytic enzyme. This bacteria produces a proteolytic enzyme due to which there is excoriation of the skin. So that is why this angular features they come. And the treatment is also specific for this one. We give antibiotic ointment, which is oxytetracycline, 1%, and we add zinc oxide. Why the zinc oxide is added? That is for the skin excoriation, because it neutralizes the proteolytic enzyme. So zinc oxide is also added there. Okay. Now this can be seen as image question also. Okay. So angular conjunctivitis, important topic. It can be seen as image question. So they will show you at an angle of the eye, there is excoriation of the skin and congestion, like, no congestion in this area, congestion only in the angle area. Then they can ask you the causative organism. They can ask you the treatment. Zinc oxide, you have to remember. Why it causes excoriation? Because it produces a proteolytic enzyme. So this is important topic for your exam beta, angular conjunctivitis. Okay, fine friends, right? Then ophthalmia neonatorum. Ophthalmia neonatorum, another type of conjunctivitis. Please come to this. Ophthalmia neonatorum. So you don't have to write much. So we can keep reading, we can keep understanding, we can keep marking. Ophthalmia neonatorum, what does it mean? The conjunctivitis which is seen within 28 days of life is called ophthalmia neonatorum, that is in the neonatal period. Okay. Now, any discharge in neonate is pathological, because neonates, they do not produce tears. So if you see any discharge in a neonate, it means it is a pathology, it is not physiological. Okay. Now, there are 4 etiologies of ophthalmia neonatorum. 4 etiologies. What are those? So, first of all, let me show you the picture here. So, this is how, if there is a baby and you see the sticky discharge here in the baby's eye, that is your ophthalmia neonatorum. Okay. There are 4 etiologies. Okay. So, sorry, you can say 5 etiologies actually, including chemical. So, first one is chemical. The second, it can be due to gonococcus. Third, it can be due to chlamydia. Fourth, it can be due to staph aureus. Fifth, it can be due to herpes simplex. Okay. Now, first of all, listen to me now. The chemical etiology is due to the chemical reaction. In olden days, 1% silver nitrate was used as a prophylaxis in baby's eye, neonate eye. But some children, they used to have conjunctivitis in reaction to silver nitrate. Okay. The use of this silver nitrate was called Creday's method. So this is not actually a heading. This is Creday's method, is that they used to use 1% silver nitrate as a prophylaxis for conjunctivitis. Some children will develop conjunctivitis due to the use of silver nitrate, which is now obsolete. Now we use povidone iodine. What are we using now? Povidone iodine by betadine. Okay. So if that is the cause, that will appear very early. within 1 hour, if you see the baby is born and they put the drop, so that will develop conjunctivitis very early. So if it is within few hours, you see discharge in a baby's eye, then high chances that it can be chemical. Okay. Now, in the other causes, you see all these are infections: gonococcal, chlamydia, staph, herpes. They used to occur, the baby will acquire these infections when it is passing through during the delivery from the mother. Okay. So gonococcus, chlamydia, staph, herpes, they are acquired during the delivery. Now, it is seen that gonococcal conjunctivitis appears within 1 to 4 days. Chlamydia usually 5 to 14 days. Staph will appear usually after 1 week. Herpes will appear usually after 1 to 2 weeks. Okay. So even if you don't remember, it's difficult to remember these, it's fine. Just when it is very early, it is chemical within few hours. And then you see earliest is your gonococcus. Why? You can imagine, because gonococcus is very, very severe infection. So it will appear early. So if you remember this point, you will be able to solve the question if it comes. That a conjunctivitis appearing within 4 days will be gonococcal. Right? Now, here few points to remember here before the treatment. Most dangerous, as I told you already, is your gonococcal. So it is very important that gonococcal conjunctivitis in a child is taken very, very seriously. Okay, it's taken very, very seriously. So how would you treat the different type of the ophthalmia neonatorum? If it is chemical, do the proper eye wash, stop the exposure to that chemical. Gonococcal is most dangerous. So please highlight this point, because it can involve cornea. So as I told you, in adult, you have to use antibiotic ointment, you have to use atropine, and you have to use systemic antibiotic also. Okay. For chlamydia, we prefer tetracycline ointment. For staph, you can give broad spectrum antibiotic. For herpes simplex, it's a virus, you give antiviral ointment. Okay, but gonococcal is most dangerous. You have to give systemic antibiotic. Okay, and which is the most common? Most common is chlamydia. So again, an important question. Okay, so let me summarize this for you. And then we can take the lunch break here. So in ophthalmia neonatorum, again, high yield topic for the exam. Any discharge seen in neonate within 28 days of the life is ophthalmia neonatorum. Ophthalmia neonatorum can be chemical secondary to silver nitrate, or it can be infection due to gonococcal, chlamydia, staph, herpes. Chlamydia is most common. Okay, chlamydia is most common, and gonococcal is most dangerous. Okay. How to treat? Chemical, stop exposure, eye wash. For gonococcal, remember systemic antibiotic. Chlamydia, tetracycline. Staph, broad spectrum antibiotic. Herpes simplex, antiviral ointment. Okay. And for prophylaxis, so I told you the silver nitrate method is now obsolete. What we use for prophylaxis nowadays, we can use povidone iodine, erythromycin, or tetracycline. Okay? So if the question is like which all can be used for ophthalmia neonatorum prophylaxis, that is povidone iodine, erythromycin, tetracycline. Okay? So this is about your ophthalmia neonatorum. We take a lunch break now, and we will be meeting in 45 minutes. That is at 2:30 to continue our diseases of conjunctiva, and then of course cornea. Okay? right, friends? So let's meet again in 45 minutes. Let's have the lunch and see you at 2:30 now. So here comes your lunch break. See you at 2:30, friends. Yes. Have a good lunch, re-energize yourself, and let's be ready to absorb more of ophthalmology afterwards. Thank you.